Dr. Sharik, it's 11:05. Shall we start the session?
I think we can get started.
Okay, sir. Thank you.
Dr. Devendra, just for your information, this is also being streamed live by YouTube. So if there are anything that later on you would want to redact and from your presentations, if there is any confidential information that you wouldn't want it to stay later on, please let us know. Accordingly, we'll edit the version and repost it on YouTube.
Okay.
Yes, Professor Abu Sufyan, we can get started.
Okay, sir. Thank you. Assalamu alaikum and a very warm good morning, everyone. I hope you have enjoyed all 5 days of 1 week AICTE ISTE sponsored faculty induction program on multidisciplinary and translational drug development with sub-theme of QBD in pharmaceutical sciences. I am Abu Sufyan, Event Lead Coordinator on behalf of Anjuman-e-Islam Kalsekar Technical Campus, New Panvel. Extend my warm welcome to the resource person, all dignitaries and faculty members to the concluding day of this refresher program. To start with, as a tradition of Anjuman-e-Islam, I will recite and translate few verses from Holy Quran. A'udhu billahi minash shaitanir rajim. Bismillahir rahmanir rahim. Ya ayyuhan nasu inna khalaqnakum min dhakarin wa untha وَجَعَلْنَاكُمْ شُعُوبًا وَقَبَائِلَ لِتَعَارَفُوا إِنَّ أَكْرَمَكُمْ عِندَ اللَّهِ أَتْقَاكُمْ إِنَّ اللَّهَ عَلِيمٌ خَبِيرٌ. The translation of these words from Holy Quran is, O mankind, we have created you from a male and a female, and we have made you into tribes and sub-tribes that you may know one another. Verily, the most honorable amongst you in the sight of Allah is the one who is most righteous among you. Surely, Allah is all-knowing, all-aware. These words from Surah Al-Hujurat that teach us to do righteous deed. It is a worth implementing in our life. Yesterday, we had very enlightening session by Dr. Muralidhara Anandmurthy on QBD using SAS JMP software, Dr. LD Patel on Six Sigma as a QBD tool, and by Mr. Bimal Kumar Kesh on QBD by using Minitab software. Going ahead, today we have very relevant session on QBD approaches in formulation and developments. For this session, we have with us Dr. Devendra Ridhurkar. I take this opportunity to introduce him. Dr. Devendra works as an expert scientist formulation and development at Nurex Pharma, Barcelona, Spain. He is responsible for development of an innovative formulation. He has over 16 years of experience and was associated with various pharma giants like Aegis, Hungary, Dr. Reddy, India, to name a few. He is expert in using platform technologies like hot melt extrusion, gastro-retentive drug delivery system, nanotechnology, and cyclodextrin complexation. He is an expert in materializing the design of experiments and quality by design. He obtained his MPharm PhD degree in pharmaceutics from IIT BHU Varanasi. He is a member of editorial board of peer pharmaceutical journals and has earned over 8 peer-reviewed papers in various international and national journals and 5 patents to his credit. He has been associated with various pharmaceutical bodies in India and American Association of Pharmaceutical Scientists. He is a member of Program Advisory Committee for Pharma Connect Congress, Hungary, and has presented his research work in several international congresses and scientific meetings. With this brief introduction, I extend my warm welcome to Dr. Devendra Ridhurkar, and I request him to start with his session. It is over to you, sir.
Thank you, Dr. Sheikh, for giving a nice introduction. Good morning, everyone. So I would like to share my presentation. Dr. Sheikh, is it visible?
It is visible, sir. Just make it full screen.
Yeah, thank you. Okay, so good morning, everyone. Today, my topic of the presentation is quality by design and how we are going to use in the formation development. So during a formation development, there are various stages. So first one is the selection of the candidate. So like the drug. So first we select the drug, and based on the physical characteristic of the drug, then we design the product design as well as the development stages. So in product development and the design stage, what we do? Initially, we do the scoping, like what will be the route of administration, like whether it will be the parenteral, transdermal, or solid oral. And once you identify it, then we need to work on how we'll optimize this process. Then the second one is the lab scale development, and that comes in the category of the process design and the development. In that, we do the process characterization, like which method, in case if we select the method that we want to develop this product by using the dosage form like tablets or the solid dosage form, then in that, which method we will use, like whether we'll use the wet granulation, direct compression, or compaction. And based on that, we need to work on the process characterization. And also, we need to find out whether the process is robust and how we'll optimize it. Once you did develop the formula and now your formula is ready for the scale up, you need to move for the production. In that, you need to predict what will be the scale up parameters and how we'll develop the control system during the transferring this technology to the production. And afterwards, you will file this dossier to the particular respective regulatory agencies. Like if you want to file this product in the US, you need to file it to the US FDA. And if it is for European Union, you need to file your dossier to EMA. And once it is approved, then we need to follow it for the continuous improvement. If we have any problem, we will work like what are the problems and how we'll training it. So this is a life cycle of the product. Now the next one is, now it's a generic era. So most of the companies, they would like to file the products in the generic because innovation, it takes a long time and also it leads to lots of money. So most of the companies in generic companies, what they do. So as there is a lots of competition, they want to file the product as early as possible. And they don't matter what will be the learning. So file first and the learning later. This is the trend in the generic industry. And when they file the product, the regulatory agencies, they review the documents, and then they found there is a lots of gaps and lots of frauds, lots of faults. And then the regulatory agency asked to make some major amendments during the review process. For example, there is a problem in the stability, like exhibit batch stability failure. If they have found that formulation is not good, they will ask for the revision. And because of that, it takes longer time for the approval. And sometimes it happens that even if the product is good, they are not marketed because it takes a longer time and they didn't approve the product. And also it is not marketed. Sometimes it also happens that you need to change the formulation once you have approved. So this is because of the improper development of the formulation in the initial stage. So now what are the root cause for this? First one is the API variability. When we select the API, we need to be very sure that we will get the same kind of API every time, like when you manufacture the batches. So there is, because API variability is the most important part in this case, like particle size, is pH solubility, is like partition coefficient, as well as its physical characteristic. The second one is the excipient variability. As only APIs can't be used in the formulation, we need to mix this API along with some excipients. Maybe it is diluent, maybe it is disintegrant. So we need to be very careful while selecting the excipient. So we should always avoid the excipient variability. Third and fourth is the process variability. Like when you use the wet granulation, then you should be very sure what will be the water quantity, which will be the equipment. and also the packaging variability, because during the stability, we need to optimize which will be the packaging, whether it will be alu or it is a blister, because based on the API characteristic, we also determine what will the packaging material. And this is the equation which shows the product stability or the product robustness is totally depend on the API, excipient, product, packaging, and the interaction. So now these are the problem, particularly formulation scientists face during the development of the formulation from initial to the commercialization and even the life cycle management. But now why FDA has initiated this quality by design approach? The first and the most important advantage of the QbD is the product is designed to meet the patient requirements. By using the QbD, we can get the consistently meet product at CQA, which is the important part of QbD. Third point is the impact of formulation components and the process parameters on product quality is understood. By using this QbD, we can easily understand what will be the impact of the formulation components. Maybe it is the process, maybe it is the excipients or different variables. And also the critical sources, and we can easily identify by using the QbD what is the variability and what is the critical source of this variability. And then if it is identified, we can easily control it. Now, this is why there is a changing environment. Before a few years, the approach was like we are generally analyzing the testing and the inspection, doing the inspection of the formulation once, and it was justified. Okay, now the quality is good, but with the QbD approach, the quality built into the product and the process by designing and based on the scientific understanding of that process and the formulation. Currently, we are following the data intensive submission and the information which is without any big picture or without any big information. But with QbD submission, we show the product knowledge and the process understanding. In current approach, the specification is based on the batch history that we prepared the batch record, and in that all the information is mentioned. But in the case of QbD approach, the specifications are based on the product performance and what are the requirements. And also in QbD approach, the process is flexible and we have a design space in which we can make a continuous improvement. In current approach, generally the focus is only, okay, we'll get the reproducibility. Even we ignore the variation and also we avoid the variation. But with the QbD approach, the main focus is on the robustness and understanding the controlling variations. So now coming on the QbD, what is QbD? So it is first introduced by the Joseph M. Juran, and he is a quality expert. So he mentioned that it is a proactive, the QbD is a proactive, systematic, and risk-based approach in the product development to predict the quality of the product. It always begins with the predefined objectives, like it is known as the target product profile. Then it gives a better control, and it will be very easy to handle the quality crisis during the life cycle of during the early stage as well as the life cycle in the complete life cycle of the product. Generally, it emphasizes the product and process understanding, process control, and it all happens because of the sound scientific knowledge and the quality risk mitigation assessment. Now, so we plan the quality into the process. But most of the people, they think it is an expensive tool. But I will say this is the myth. Then what is the fact? The fact is, the QbD is a tool that makes product development and the commercial scale manufacturing is very simple. So when you implement the QbD in your product development and the commercialization, even the scale up will be very easy and very simple. Then, and finally, also it saves the money. So in the next slide, we'll see how it will helpful in the product development, how it will save the money, and how it will make the process simple. So how QbD will help to improve? So QbD will help to ensure higher level of assurance of product quality for the patient. How? With QbD, you will get the improved product, good process design, and a good understanding of the product. By planning quality at first place to avoid quality crisis. And the third one is monitoring, tracking, and training of products and the process. So it will help to improve the product understanding. QbD also helps increase price control and timely completion of the development activities by increasing the efficiency, by implementing the QbD execution, which is improved time to market. It also minimizes and eliminates potential compliance action by the regulatory agencies and the less validation burden. As per the regulatory requirement, we need to validate the 3 validation batches we need to manufacture in the production. But with QbD, we can minimize this 1 or 2. Then continuous improvement over the total product life cycle. Once your formulation in the market, you can continuously improve by using the design space. And because of that, you can decrease your product recalls and the rejects. And also the post-approval changes and efficient regulatory oversight can be minimized. So the next step is how we can implement this QbD in our formulation development. So there are 5 steps. When you decided, no, I want to develop my product by using the QbD. So how to implement and how to identify these 5 steps? The first step is identify the target product profile. So it is known as the drug product objective. So discuss with the team and then decide based on the physical characteristics of the API, what is your requirement, find out, okay, what will be the target product profile, like the dissolution or assay or impurities. And based on your target product profile, define the CQA, the critical quality attributes. Once you have identified this TPP and CQA, then move to the next step. Then identify the process, and related to this process, what are the critical material attributes and what are the critical process parameters? Like if you found that the process is wet granulation, then what are the variables that should take care? For example, the addition of the water speed, the RMG speed, impeller speed, chopper speed, then drying temperature. These are the different processing parameters. And during that, you need to find out the risk management by using the risk elimination matrix, that REM, and also the failure mode effects analysis. Once you perform this stage 2, you need to move to the stage 3, or step 3. Carry out some response surface optimization by using some experimental design. You can use the DOE in this step, and then explore the CQA at high, medium, or low levels, and also prepare some formulations by experimentally prepared according to the design matrix. Once you have a robust formula, then you can modelization and validation of the QbD methodology by using the response surface methodology, that is RSM. You can use 2D or 3D response surface plots, and also you can use the multivariate chemometric techniques in order to validate the QbD methodology. Once you have a scale-up formula, you have validated the QbD methodology, finally you can transfer your process for the scale-up and the production. So in this case, you need to find out, or you need to work on the various product and process parameters, what should be evaluated, and the results should be compared with the predicted, like the theoretical and the practical. And to confirm the QbD performance, the product or process should be scaled up. Now coming on the next slide, it is the key aspects of the QbD. So there are 7 approaches for implementing the QbD. The first one is to identify the TPP. The second is CPA. I will explain it one by one in the next slides. The first is TPP, CQA, CPP, then risk assessment, and the design space. So risk assessment and design space are very much related to each other and they can interchange. Then design space to control strategy and the life cycle management. So coming on the first, target product profile. In TPP, we generally use as a tool for setting the strategic foundation during development in the context of goals. So we find out we've set some goals, like how we'll use it, what is the safety of this formulation, what will be the efficacy of this product. And this tool also can be used by formulators as a quantitative surrogate for aspects of clinical safety and efficacy. And the main importance of TPP is it plays a central role in the entire formulation development process, because TPP is initially we decided, like I earlier told you, what will be our dissolution pattern. If it is modified release, then what is the dissolution hours, like 12 hours or 24 hours, what will be the safety, what will be the efficacy, the pharmacokinetics. So this comes under the TPP. And there are different components which are generally related to the safety, efficacy, and identity, as well as the purity and the potency. So which are these basic factors? The first one is the critical, and there are two factors. One is the critical and non-critical components, and another is the fixed and variable components. In critical, it is like assay, contain uniformity. Non-critical is appearance. Of course, appearance is very important for the elegance and for the patient compliance. But the critical is like assay. If your assay is, ideally the limit is 95 to 105, but if it is below 95, then the product is not good, or it is fails in the assay. So that's why it is very critical. The second is fixed and variable components. In fixed elements like dosage form, strength, and the variable elements, that the tablet weight and the assay. This is a simple example of the TPP when a formulated develop and immediate release solid dosage form. So what should be the TPP? For example, the dosage form of the reference product is tablets. Then what will be the route of administration, whether it is oral or parenteral? Then what is the strength of this dosage form? If it is a tablet, like for example, 500 milligram, the minimum dose and the maximum dose is 3 grams. Then what are the physical properties you should be considered, like its appearance, if it is a tablet, like weight, friability, hardness, disintegration. And as we need to prove the pharmacokinetic by using the bioequivalence study, we should perform the dissolution and aerodynamic performance, that is known as the pharmacokinetic characteristic. Its identification is assay, contain uniformity, impurities. We need to check if we are using any residual solvents. If we are using any organic solvents for coating or the granulation, we should determine the residual solvents. As per EMA, the requirements is to check the microbiology, then the release and the delivery of the drug system, whether it is what kind of drug delivery system it is, whether it is osmotic. Then based on that, you need to perform the kinetic study and find out the drug release mechanism, whether it's a first order or zero order. Then its pharmacological characteristic. Finally, it is labeling and also the packaging and the pharmaceutical elegance in order to get the better patient compliance. The next is the CQA. Generally, CQA is a characteristic that should be within an appropriate limit or distributed to ensure the desired product quality. So CQA is a very important part of the QBD. CQA includes the parameters that are likely to change based upon variation in raw materials. Like for example, if you are using the HPMC and you are developing a modified formulation, so viscosity is very important in that case. Every company, they have some specification. Okay, for viscosity, for example, 100 to 200. But you should need to be very careful while selecting the polymers based on the viscosity, particularly for the modified release formulation. If they change the specification, then your product release will be changed. Then another factor is the assay and the contain uniformity. Generally, the CQA is identified based on the risk assessment as per the ICH guidelines Q9. And if you have a prior knowledge of the product and the clinical experience with the specific product quality attributes, it is very useful to making a risk assessment. It will also include the relevant data from similar molecules and data from the public domain or from the literature search. Then what factors are generally affected by CQA? First one, the input and the in-process materials, the properties of input and in-process materials. For example, The purity, potency, and surrogate for viability are very critical. The levels of critical depend, like if there are different manufacturers of API, that will also very critical for the CQA. How the TPP, and this is a correlation or the relation between the TPP and CQA. So how the TPP components and the CQA are related. So like dosage form, route of administration, and the CQAs are in the right side. You can see the CQA like assay, impurities, content uniformity, and the dissolution. So it all depends on the TPP. The third one is critical process parameter. In CPP, these are the parameters which impacts a lot on CQA, and also it affects the production process. So it is very important. It must be monitored and controlled within the predefined limits to assure the product meets its predefined quality attributes. And to demonstrate the reproducibility and consistency of a process, there is a capability index, and it should be calculated if you want to determine the CPP. So the calculation is like upper limit of specification minus lower limit of specification, and there are 6 standard deviation. And then how will identify the CPP is good or not? If the CPK is significantly greater than 1, the process is defined capable. So the benchmark is 1. So ideally it should be always more than 1. And if the process capability is lower, follow the 5-step procedure to progressively reduce the variability of the process. Then you need to follow this process to reduce the variability in your formulation. So step 1 is define your task. Second is measure your potential. What are the target areas? Then analyze the problems, improve your process, and finally identify the critical issues. Try to improve it and then control it. So these are the 5 steps you need to follow in case if you have CPK greater than 1. The 4th QbD factor is risk assessment. What is this risk assessment means? It is a science-based process for the assessment in which we control, communicate, and review the risk for the quality of the product. And generally, it works by using the identification of relevant quality process CPPs, eliminating the risk and resulting in the desired TPP. But why it is important to the risk assessment? Because it is very important to identify the relative risk levels at the beginning of the product development. Also, it is very important to prioritize the limited development resources, the document the decision-making process throughout the development, to assess the needs of additional studies for scale-up, because finally the product should go in the production. So for that, you need to do the technology transfer. So if you identify the risk initially, we can easily understand what will be the scale-up and technology transfer problems. To identify appropriate specifications, CPP, and the manufacturing controls. And also, finally, the important thing is to decrease the variability in CQA. And generally, a formulation scientist do the risk assessment for the formulation. In case of formulation, we generally do the risk assessment for the starting material properties of the API or the excipients. Then manufacturing process, we always make sure the process is robust and find out which method is the best for this kind of formulation. And then how identify? There are the certain steps for the risk assessment. First, list out all components of process that is involved in the formulation development. Make a process flow chart. It will be easy. Then it will be easy to find out the problem or the specifications, or to control the strategy. Identify all potential failure modes for each item with risk query. Maybe what might go wrong. do risk analysis and the risk evaluation. There are different tools of the risk assessment. Like we can use the flow charts, check sheets, process mapping, and cause and effect, the Ishikawa diagram. We can do the FMEA analysis, that's failure mode effective analysis, by using systematically systematic identifies, prioritize, and estimate the risk and the prevents failure. Also, we can use the fault tree analysis, which is very good, which is structured, graphical, quantitative assessment tool. And also it is an excellent tool for evaluating multiple factors affecting product quality. Also, there are various ways of the risk assessment, like failure mode, effects and critical analysis, hazard operability analysis, risk ranking and filtering. And we can use some statistical tools for this risk management. The fifth one is the design space, in which we can work and to make the robust formula or to design a formula which as per your requirement of the TPP. So what is TPP means? What is this design space? The design space is a multi-dimensional combination and interaction of input variables. So what are these input variables? It is the material attributes and the process parameters, like the physical properties of the excipients in the API, along with the process design. For example, process parameters, as I told you, maybe if it is for the weight granulation, the important process parameters are the granulation step, the drying step, the milling, and the compression. And if it is a coated product, the coated. This is the process parameters which are important for a solid dosage form. Then critical concept behind the design space. It demonstrates to provide the assurance of quality of drug substance or the drug product. Here are more objectives in drug products. So it provides the quality of the formulation will be robust. And once the design space is approved, regulatory post-approval changes requirement will be very simplified. So if you include the design space in your dossier, then after, like you want to change the formulation after the approval, then it will be very easy to modify or simplify the changes in your formulation. And it's a regulatory flexibility you can operate in the during this design space. So these are the advantage of design space. Then how we can utilize the design space? We can use extensive monitoring during the development of enhanced process understanding, and also we can use the science-based control during the manufacturing in order to implement the design space. Sixth one is the control strategy. A control strategy means a planned set of controls derived from current product and process understanding that process performance and product quality. Also, it is established by risk assessment that takes into an account the criticality of the sequence process capability. And it has the ability to evaluate and ensure that the quality of in-process and our final product is robust. What are the criteria we should include when we want to follow the control strategy? Try to control the raw material attributes like drug substance, excipients, packaging, the equipment operating conditions like processing parameters, finalize the specification for final dosage form, then the methods and frequency of monitoring and the control, the specification, procedure controls. We need to monitor or include the elements like process monitoring, characterization, and the dissolution testing and the stability testing. The last part of the QbD is the life cycle management. As we already filed the product and now it is commercialized, but there are several ways to control the life cycle. And with the QbD, it will be very easy to monitor the life cycle management. So what we'll do in the QbD paradigm? It is the process changes within the design space, and we will not require any review or approval, post-approval changes from the regulatory agency. This is the biggest advantage of the QbD. And that's why the process improvements during the product life cycle with regard to process consistency and throughput could take place with the fewer post-approval submission. So it will be very easy if you want to make some post-approval changes. In addition to that, it provides regulatory flexibility. It provides a good understanding of the manufacturing process as well as the risk assessment as per the ICH Q9 guidelines. And also it helps for the continuous improvement of the essential element as per the model quality systems. Now coming on the tools, there are various tools that can be used for the QBD. The first one is the design of experiment. The design of experiments is a well-structured and organized method to determine the relationship among the factors. Like factors means like the CPP and like processing parameters related with the drug release or the assay and impurities. DOE also helps to study the effects of interaction of multiple factors at a time. What is the application of DOEs? The first is scope out the initial formation of process design. It also helps determine the design space, including the multivariate relationship. And also finally, it is very useful for the optimization and the process scale-up. The advantages of DOE is it is very useful technique for screening of variables by considering the impact on drug products CQAs. It offers a returns 4 to 8 times greater than the cost of running the experiment in a fraction of the time. And also it helps in getting the robust formula. There are different methodologies in order to run a DOE. The first one is to choose the experimental design. Maybe it is a full factorial or a d-optimal. We can conduct the randomized experiments. This is the step of the DOE methodology. Then third, you can analyze the data, and then we can create the multidimensional surface model like 3D or 2D. So like on the first, like how to choose the experiment design. For example, I will explain it by one example. Like if you want to develop a modified risk formulation based on the HPMC, then we need to decide the different levels of concentration of HPMC, like low level. If you want to go for the 3-factor design, try to decide the low level, medium level, and the high level of polymer quantity. And then you can plan different variables, and you can perform DOE by using softwares like Design Experts. Generally, I use the Design Expert. These are the softwares that generally has been used in the pharmaceutical companies. The next one is the process analytical technology. This is, in short, it is known as the PAT. So what is PAT? It is a system which is for designing, analyzing, and the controlling manufacturing through the timely measurement during processing of first critical quality and performance attributes. The second is raw and in-process materials. Nowadays, most of the pharmaceutical companies are working on the continuous manufacturing. So it is good technology to use it at line, online, or inline measurement to determine the water content or to determine the assay during the manufacturing. Then what are the objectives of this PAT technology? To enhance understanding and control the manufacturing process with focus on, in, or at line. So now the focus has been changed due to the regulatory perspective. In the continuous manufacturing, we need to analyze most of the contents, like during the granulation, you need to control the weight granulation, the addition of the water rate. So by using this PAT technology, you can analyze online. So you don't need to stop the process, go to the lab, and then perform the water contain or assay. But with this PAT technology, it is very easy to analyze online. What are the advantages of PAT? First, the most important advantage is it reduces the release testing of the product. As I told you that you need to stop the process, or after the completion of the process, you need to take sample and then go in the laboratory and run the HPLC. But with the PAT, it is not required. You can use the NIR technology. Just in 1 minute, you will get the required assay or water contain or the different kind of testing. and it offers a non-destructive online analysis of a process. It can be achieved the goal of real-time release testing. It reduces the inventory and also lower the laboratory cost. Additionally, online or at-line PAT sampling allows for an increased sample size. And also it assures the greater quality measurement. These are some PAT tools which I generally use in the tablet manufacturing. For example, if we are dispensing the material for the formulation development, so during the dispensing stage, I generally use the NIR or the Raman, and it generally used for the identification of raw materials. Whether if it is HPMC, okay, I can identify, okay, this is HPMC. So previously, you don't need to find out, okay, I don't need to perform the analysis in the lab. And during the wet granulation, I generally use the NIR to determine the moisture distribution, how much quantity of water I added, whether distribution during the granulation is uniform or not, and how much quantity I need. So I can easily identify during the wet granulation by using the NIR technology. During the drying, it's also the same, the NIR, where I can determine the moisture content. During blending, NIR also is useful in the blending to uniformly to determine the blend uniformity. For the compression, there are two different techniques. One is the strain gauges and the NIR. With strain gauges, I can easily determine the compression force. And if I want to determine the continuity, I will use NIR techniques. And recently, QbD has slightly modified, this is the added segment that is known as the formulation by design. This is one segment in the QbD that now we can develop the formulation by using the formulation by design approaches. So this is the recent QbD-based paradigm. It is exclusively applicable for the development of pharmaceutical dosage form. It includes the product and process understanding, and these are the twin keystones for the formulation by design. It requires the holistic envisaging and the formulation development. It predicts the impact of quality CQAs during laboratory scale-up and production of exhibit batches. And finally, it gives a robust and stable drug product. So this is all about the QbD. And I hope you got more clarity about the QbD in the formulation development. Thank you. Thank you for listening to me.
Thank you, Dr. Devendra, for giving very informative and fruitful session. Going ahead, we will take up a few questions of the participants. I request our participant to post your questions in a chat box. Meanwhile, there are already few questions. We'll take up those questions first. So Mr. Tushar Sarwan is asking about how the control strategy for the product and process be applied separately. Can you give some case study?
Yes, when I was working on the continuous manufacturing process, so I used the design expert software in order to find out the processing, and it was continuous manufacturing based on the hot mill extruder. And the idea is to perform the weight granulation. So I use the software in order to determine what will the water quantity and what will the screw speed. So, and based on these processing parameters, I optimize. I got the good compacted material, and it's better than the weight granulation process that generally we used to follow in the weight granulation RMG, the rapid mixer granulator or the high shear granulator. So then I identified the hot mill extruder is better in order to get more compacted material.
Okay, I hope it's clear to Mr. Tushar. Next question it is by Mr. Sahil Arora. Is asking that any optimum method for fixation of optimal range for critical parameter in DOE studies?
Yes, like for example, if we are developing a product based on and modified this formulation based on the coating, your tablet says immediate release. And in case if you are using the release coating in order to get the 24-hour drug release profile, as the release is water insoluble, you need to add some performer. So maybe like water soluble polymers like HPMC or Poidon. In that case, you need to identify what will be your performer quantity or the ratio between the ethyl cellulose or the surulese to the polymer. And in that case, it is very useful. The DO is very useful.
Okay, thank you, sir. Next question again by Mr. Tushar Sarwan. He's asking that which is the best software to control manufacturing process online? Have you used or came across, especially in continuous biomanufacturing? And he's also asking that what is the level of model is suitable and best, like MVDA, NN, deep learning in the pharma and biopharma industry?
I'm sorry, but I didn't came across in the manufacturing. Like, yes, I use the NIR, but this is for the continuous manufacturing, the Raman spectroscopy, but not for the manufacturing in the production.
Okay, 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 sir.
I'm related with the small molecules.
Okay, okay. He has also asked about which is the best software to control manufacturing process.
I think all the softwares are there. There are lots of softwares, and it depends on your requirement. So I don't have any choice of the specific softwares.
Okay, okay, okay.
So almost all softwares which are generally used in pharmaceutical companies. They are all robust and they good, very good determinations.
Okay, sir. That's it. I think these are only the question. If there is no further question, then I think we'll conclude our first session of the day one. Let's wait for a few seconds. We'll see whether there are questions coming in. It seems there is no further question. So thank you, Dr. Devendra, for very insightful session on QBD approaches in formulation and development. To summarize, you have discussed in very detail about different practical aspects of TPP, CQA, CPP, risk assessment tools, design space, DOE, etc. Your session has clarified many important aspects of the QBD. So on behalf of organizing team and the participants, my sincere thanks to you, sir, for giving your valuable time and sharing wonderful aspects of the QBD. Thank you, sir.
Thank you, Dr. Sheikh.
Thank you, sir.
Right.
So,
yeah.
So now let's take a short lunch break and we'll resume back sharp at 1 p.m. for interesting panel discussion, which will be led by Dr. Bhupender Singh Bhoop.
Good afternoon, madam. Dr. Sharik here. Welcome back. Dr. Vandana Patrawale. Dr. Shirsagar is already here. We're just waiting for Dr. Bhoop. He had some power issues at his home, so he'll be joining in a couple of minutes. I think Professor Sufyan, you might want to make Madam co-host. Good afternoon, Madam. Dr. Sharik here. Welcome back. Dr. Vandana Patrawale. Shirsagar is already here. We're just waiting for Dr. Bhoop. He had some power issues at his home, so he'll be joining in a couple of minutes. Professor Abu Sufyan, you might want to make Dr. Vandana Patrawale host. She's okay.
Now she is the co-host.
Okay, got it.
Dr. Vandana is co-host.
Good afternoon, sir.
Good afternoon, madam. Good afternoon. Welcome back, madam.
My pleasure.
Our participants have told that we had a rock star session of Dr. Vandana Patrawale.
That's their goodness, I would say.
No, it was really well received. Why? I'm being echoed. I'm being echoed now. I'll log out and call out.
Good afternoon, sir.
Good afternoon. Welcome, Dr. Bhoop.
Sir is on mute.
So you need to unmute yourself.
Okay. Just a bit.
Good afternoon, Shirsagar, sir. You are on mute.
I think as of now, the participants have been muted, so we'll have to make him co-host to get speed. Just a second, sir. Professor Abu Sufyan will be making you co-host, so that way. Yeah, you can unmute yourself now.
Okay. Good afternoon, everybody.
Good afternoon. Good afternoon. Welcome back once again for
Yeah.
Nice to have you back, all of you, sir. Thank you so much for taking the time out. Madam is also here. Dr. Bhupendra Singh is also here. Dr. LD Patel has also joined. Abu Sufyan?
No, sir, not yet.
I think I had messaged him that you might get a little delayed, so maybe he'll join in a second or so.
Okay, 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 sir.
Dr. Bhoop, we had messaged all of our participants that we'll be starting at 1:10. So we'll continue with that time frame. Is that okay?
Just a minute. Just a minute.
Okay.
Okay, you have sent some questions beforehand. I'm just forwarding it to another mobile of my better half to read them, because this mobile is busy in communicating with you all.
Okay, not a problem, sir.
In fact, I'm currently powerless.
Okay.
There is no power, at my place for the last 15-20 minutes. No Wi-Fi, no internet. So I'm unable to make use of my laptop. So
no issues, sir.
My mobile is, I think, adequately charged.
Good, that's good.
I have got more powerful mobile of my better half with me to read the questions.
That's good to hear. That finally she's come to your rescue, as always in life. As they say, life comes to rescue.
Only mobile is not powerful. Everything is powerful for the better half.
The support of the better half is powerful.
The support is there. There was a joke that was circulating around. He said, if the man is the head of the house, who's the wife? They say the wife is the neck of the head, which turns the head in any way they want.
Very nice.
That was a joke of the pandemic, I guess. I awarded it.
In fact, they have to complement with each other. Then only the life goes on.
Pandemic has been a true test of their patience when we are all at home all the time. So it has been a test of their patience, I must say. I think my wife says she's been praying in the night at late hours so that this pandemic gets lifted and I go back to my office doing what I should be doing.
So, I read out. I think we are geared up. 1:10.
Yes.
It's okay, Hannah?
Fantastic. Yes, sir. Surely.
So,
some of the questions that you had forwarded, should I forward it to other members or how do you?
जो मैंने अभी आपने जो भेजे हैं ना, जो उसको segregate करके मैंने आपको भेजा है.
तो उन्हीं को मैं सबको भेज दूं, if you permit. I think we lost his connection.
Sagarji and Dr. Vandana. So I was just segregating because some of the words were joined, and I've sent it back to Dr. Sharik. He's sending it to you all.
I'll do that, sir. Just a minute.
So the first question, I think, good afternoon to all the participants and all the panelists, particularly Dr. Vijay Sagarji, Dr. Vandana Patravale ji, and I think Dr. Muralidhara is also there.
Absolutely. Yes, sir. He's also on call. Yes, he's here.
So good afternoon, Dr. Muralidhara as well. We, Dr. Vandana Patravale, and we were on another IPA platform. And we just left to join this, very important panel discussion. So I have a slurry of questions. Number one is correlation of dependent variables with independent variables, how to get using regression equation. Very simple. In fact, this deals with the fundamentals of regression. So if it is a univariate data, so typical equation y is equal to mx plus c or a plus bx, comes to your rescue. But invariably in the DOE part of QBD, we get a multi, factor equation, which is called as multilinear equation, and which is an outcome of multiple linear regression analysis. And the equation is something like y is equal to a plus b1 x1, b2 x2, b3 x3, and bn xn, like that, if there are n variables. Normally, as we had been talking, normally 2 to 3 variables lete hain hum optimization mein, sometimes in certain remote cases 4, while in case of factor screening, it could be any number. It could be 11 number, it could be 15 numbers, 17 numbers. So that equation is developed thanks to the computer software based upon these statistical algorithms. It's generated by that, and it not only generates the equation, it also diverges the statistical discernment, statistical significance of each of those coefficients, whether intercept coefficient or the individual gradient coefficient. That's a plus b1. b1 is the gradient, b2, b3 are the gradient of each of the factors, while a may be the intercept. So due to paucity of time, I'm not elaborating. I could have passed on this question to Dr. Muralidhara to elaborate in a different perspective, but in fact, there are so many questions, and within a span of less than 1.5 hours, because their valedictory session is also scheduled at 2:30. So and we will be delighted to hear Dr. Ajaz's hand. So I take the next question. Next question is how to carry out risk assessment in during QBD studies. Already I have touched upon during my presentation. I do not know, other speakers must have also dealt with this part of exercise. Is, I think, being carried out by all medium to large scale pharma companies across the country and across the world. They may not be carrying out the complete QBD exercise, DOE, or risk assessment is being carried out, that which are the high risk factors. And for that, people from all, departments come together, particularly the formulation development and analysis department. These two are considered as very essential. So they come together and they sit down and they harp at which are the high risk and low risk factors from likelihood point of view, that's frequency point of view, from severity point of view, and in certain cases from detectability point of view. If you are taking detectability, it means you are roping in the technique like failure mode effect analysis. If you are going in for a simpler technique like risk estimation metrics, then you need only likelihood and severity. And the high risk factors, that's the red ones, are to be taken care of. Please open, please. Okay. And with respect to the third question, with respect to third question, that's in order to fully grasp the concept of QBD, what sources can we look into? Please open. Please open. Fully grasp the concept of QBD, what sources can we look into? So there is no single book which I can refer. So Pharmaceutical Experimental Design by Lewis et al. Is the comprehensive book, but that again contains various chapters. Pharmaceutical Quality by Design by Lewis et al. By Elsevier, I think Elsevier. But I have already passed on very comprehensive articles to Dr. Sharik to be made available to one and all. And those articles, although the web portals tend to sell these articles at exorbitant cost, $140 to $220, each. They are very voluminous articles, highly sought after articles. But since I am the principal author, I can share it as for academic gift for academic purpose. So you can make use of those as the free repertoires. And also I passed on 2 very important book chapters in pharmaceutical product development book by Dr. N.K. Jain. So those chapters should also enlighten you. How can we implement and how to apply quality by design to medicinal chemistry field? I pass it on to Dr. Vijay Sagar or anybody else, who can answer. I think this question could have been answered by somebody who's dealing with the API manufacturing. Medicinal chemistry, I personally feel this question is quite wide and relatively, I'm please pardon me, quite wide and wild both, because medicinal chemistry is very huge kind of domain. And probably if he is thinking about the API synthesis, yes, it can be applied to API synthesis and various, again, process parameters and material attributes are to be delineated and optimization is done, particularly to improve the yield of the API. And, the organizers tried to rope in some of the API, manufacturing QbD experts like Dr. Gautam Samanta, but those were not available. But those experts can highlight and some, references shared by them can surmount this problem, because I don't think neither Dr. Vandana, Dr. Vijay Sagar or anybody else amongst us is
Dr. Bob, we can just add from the route of synthesis point of view that this is one of the common things which is done in the API industry today, that you have multiple routes of synthesis and there is a risk assessment which is done to find out which route is the best. And in order to do the risk assessment, it starts right from the availability of the vendor to supply the starting key material, and to what extent you can do the backward integration, because you have to minimize your dependence on the supplier. So the API industry, which is able to go as much back as possible, at least N-1, N-2 from the key starting material. And the key starting material is the one which we consider as the material which has got significant fragment of the final API moiety. So that's what the key starting material, and customarily it is not one key starting material, there can be multiple key starting materials. So since we are all generic players, a lot of routes of synthesis are available. So a lot of materials which are available. So the common thing which is done by the API industry is to find out what is the best route of synthesis considering the cost, quality, and the techniques and the PAT tools which are available at your end. So that's what I will like to add. Maybe Dr. Vandana will like to add something more to that.
Before Madam starts, I'd just like to welcome Dr. Aldri Patel also to the panelists. Welcome, sir. Welcome.
Thank you. Thank you.
Over to you, Madam. Yes.
Yes, so I feel that principles of QBD will still remain the same. So majorly, if design of experiments is what you are aiming in your planning, you need to identify which would be your reactives, which are the concentrations which will take it as independent variables, and whether it is a purity or yield which you are looking as a dependent variable or the output factor. So I think once that is very clear in your mind, principles of QBD almost will remain the same, and you need to select a particular design of experiment and move ahead, because that is the main thing. And that I can say because, my husband being president of Aarti Group of Industry, which deals with a lot of chemistry part, that is what they are doing majorly. And another aspect, of course, is, as Professor Bhoop earlier also said, analytical QBD, which forms a major thing here.
Hello, good afternoon.
Yes, good afternoon.
Sir, I'm summarizing something.
Yeah.
It's common, but I think for students and for new participants, it is essential. First of all, my participation presentations is dedicated to all those frontline pharma professional and colleagues who, while fighting the menace of COVID-19, sacrificed their life for humanity services. I pray to God for their heavenly peace. Going to our subject, QBD is very excellent approach. When it was not, we have seen the status of the industry and academics also. So definitely it is an excellent approach. But looking into the Indian environment and industries, we know that pharmaceutical manufacturing involves API, as sir has also told, excipients, equipments, qualified personnel like BPharm, MPharm, some BSc also, technician, engineer technicians, and trained worker. So all are to be coordinated. And what may be the hurdles or what is actual status that I'm telling? Now I'm summarizing.
Yeah. Thank you, Dr. Ali Patel ji.
One more thing, sir, just a minute.
Yeah.
I'm telling. Pharmacist, the working pharmacist, they detest almost in every industry. I'm not criticizing, sir. And if there is if the feeling of anyone is hurt, I apologize for that. That they are not reading. They are not updating their knowledge. They go at 6 to the industry and return at 8:30. They haven't the time to read the new things. No doubt, they may have feed up from their juniors, seniors, or others. So that is one of the lacuna. And second things, we know that formulation is a different thing and manufacturing is a different thing. Manufacturing involves technology and instruments. And the technology engineers, they are not basically known of the pharmaceutical processes, fundamentals of the pharmaceutical processes. So that is one of the lacuna. And as a consultant in pharma industry, we see that we have to also train to the engineer technician that how probability is affecting these parameters. They are not knowing the basic of parameters. They know only the techniques and methodology. So that is also involved with QBD. That is one thing. Workers, sir, everywhere in every industry, workers are given more importance than the pharmacist. Yet, I'm not criticizing again, but what I see, yet they are to be trained. They are to be trained according to the product. A company may have 100 products, and in a month, they may be manufacturing more than 10. So what are then 10 products? What are the detailed sub-steps, main steps, and what care should be taken? That is to be trained. But that is not time with the industry, sir. Again. So in all, if I say that there should be one coordinator, leader like Toop sir, which coordinate everyone, pharmacists, technicians, workers, and management. And then we may have fruit of the QBD. Regarding my topic of Six Sigma, sir, it is not known to even pharmacy peoples. Forget the technicians and other things. So it is very difficult to have implementation and implication of Six Sigma, sir. Okay, thank you, sir.
Sir, you are on mute.
Boop, sir, you are unmute, sir.
Yeah. Thank you so much, Dr. Patel, Dr. Sheesh Sagarji, and Dr. Vandana Patravale for making things vivid. Certainly, your point is very well taken, Dr. Patel, that much needs to be done. What to talk of understanding Six Sigma? So we are, quite away, quite distant from understanding the basic fundamentals of today's pharma development. And next question is, because there are, galaxy of questions, how can we implement QBD in biotechnological product development? I think there is no better person amongst us other than Dr. Vandana, who can answer this, because the only aspect I understand with respect to biotechnological aspects, that most of the parameters, that's critical material attributes, which are selected in case of biotechnological products, they are not, quantifiable per se. And so, in case of synthetic drugs or the normal excipients, we tend to get critical material attributes. Of course, critical process parameters are quite analogous, but critical material attributes are different. That's why it's a big challenge because that's why we do not get available products, but biosimilar products in case of the biologics. So biotechnological products, we have to compromise with respect to protein drugs, with respect to other biotechnological products. I think some light can be thrown by, legendary scientists amongst us, drug delivery scientist, Dr. Vandana Patravale.
Unfortunately, my experience also in that direction is not much, except for taking the biotech originated API and then formulating it to dosage form. But there again the procedure would remain the same. But when we talk about making a biosimilar using QBD approach, it's a total different ballgame. And as very correctly said by Professor Boop, one needs to do the chemical characterization in a much elaborate way when the fermentation is still going on. So there, the major emphasis has to be given to media optimization. And it is that media which will overexpress or underexpress a particular, moiety which you are interested in. So for media optimization, as you know, again, it will go back to which are the ingredients which are being used. What are the external factors? So when we talk about, of course, the ingredients, the material characteristics come. And since they are now large molecules, we need to pay a lot more attention to aspects like polymorphism, their crystal structure, whether the CD technique, circular dichroism can be used as an inline tool to monitor them. Coming off the processes, of course, it will remain the same, whether it is a temperature, whether it's a pressure, or anything else, stirring rate, etc., which can play a very important role in the process manufacturing stage. So I would say that understand that particular, again, though it is biosimilar, I can call it as again API, understand it really well with respect to whatever is available in the patent domain. Whatever characterization has been done, the literature, understand it well and then move ahead. But majorly, again, what is being done at ICTE, since they are into now, there's a group who's very actively involved in biosimilars and transferring the technologies to institutes like Serum and even Reddy's. So they are looking at these aspects very critically.
I think this is a very contemporary and futuristic concept, and I think next era tends to belong to biotechnological products.
Doctor, I may like to add something.
Sir.
Based on whatever little experience I had of biotechnology products and also with Biocon and training on some issues. I think the two major challenges which are there in biotechnology, which are significantly different than the small molecules, is that here we are dealing many times with the living organisms. And these living organisms are also having their own moods. Sometimes, in spite of providing the best of the things, they say we are tired, will not like to grow, will not like to ferment. So no amount of statistics is going to tell you what is the best mood and what is the best environment for these organisms to grow fast and ferment fast or produce fast. And secondly, as I think already said by Dr. Bhu, non-availability of the innovator product. Because in small molecules, it becomes much an easier job that you have got a reference product, you have got a reference molecule. But here, you are trying to make a similar product. Now, to what extent it should be similar, 90%, 95%, 98%, that similarity is something which is a discretionary aspect. So I think while applying QBD for biotechnology, these two things are the very significantly different aspects which ought to be taken into consideration. So when you are applying DOE and all that, we have to really see how much statistics is going to throw the light on all these variables which are significantly different than the small molecules. I think that's the addition from my side.
Thank you, Dr. Vijay Sagarji. I personally feel it's a niche area in itself.
Yeah.
So much has been already shared by Dr. Vandana and Dr. Vijay Sagarji. Because of the biological origin, there are so many hiccups involved in streamlining the QBD implementation of on biotechnological products. But it's a different area. It's entire conference can be held on QBD of biotechnological products, and experts can be called. But that gives insight to many other organizers that this could be a field of topic of further discussions. Then other questions which are being asked. What is the difference between, very interesting, I would like to diverge on the same, between QBD and design expert. Is Dr. Muralidhara available? QBD and design expert, very interesting.
Okay, so I'm going to switch on. I was just muting.
Sugar, I nahi pucha QBD and GMP.
No, I was just muting on my video because I just wanted to listen to all of you. At the same time, I'm in front of Himalayas. I just want to bend down and say I'm going to listen to you people and learn.
आपकी आवाज feeble है, Dr. Murali.
Is it clear now, Professor?
हाँ, maximize कर लीजिए.
Okay, so that's a great question. So design expert is a technology enabler. I would call that's a tool. Any technology can help you to perform a lot of statistical activities. There could be market and available in market there are softwares like Minitab, Jump, Stata, SPSS, open source tools like R, Python, anything you can leverage. And so essentially the technology should help you to perform faster and quicker. Keep that in mind. So Design Expert is a company and it makes softwares in the area of design of experiments. Likewise, Jump also makes a lot of, SAS also makes a lot of tools. One of them is Jump. So are they synonymous to each other? I'm not sure. So this is one of the softwares I would say. So use any kind of softwares which can help you to perform faster and quicker. And that's what I would put that across. So apply statistics to minimize your risk of making a bad decision or whatever you call it as informed decisions. I would put that across from a data science standpoint.
Yeah, QBD is an approach, and Design Expert is one of the software which takes care of only one aspect of DOE, one aspect of QBD, that is DOE. I think GMP tends to take care of QRM as well, quality risk management in part as well. So is Minitab, but Design Expert takes care of only DOE part. But somehow, since it permeated into Indian circles very early, so it has become synonym of QBD, design expert. But it's not QBD; it is DOE, only DOE.
Agree with you. I think QBD is
one more thing.
Yeah, G.
One more thing, sir. To Murli, sir.
Sir.
Almost.
Murli, call me Murli, please. Don't
Yes. I'm speaking in short. In all software work on the principle of LMS, least means square error. LMS, least mean square error. Error means difference between xi and x. Xi and x. It's square and minimum square. Least mean. So where it is least, that is good. And we say in term of correlationship coefficient also, R square also somewhere we are using. So almost I used all that. JMP also I'm using Minitab also, even DOE also, every and in principle they work on this.
Minimization of error.
Yes, minimization of the error. What is actual and what is your estimated?
Maximization of fitness.
Yes.
I would add another point to it.
Yes, sir. Yes, sir. So I told to you, sir.
So from a
so that we can learn more.
No. So from a data science standpoint, when we talk about data science or application of statistical techniques for informed decision making, two perspectives we always look. One is the explanatory research we use it, and then the predictive research. In predictive research, we are least bothered about learning many things. We just want to predict. Okay, so whether my whether my variable is to what extent I can forecast and predict. I'm least bothered what are the interactions. In explanatory research, where you also want to understand the process, and that's the best part of QBD is you want to understand better before you go ahead and predict something. So that's where the life science stands. So explainability is a big component in life science compared to what I learned in retail sciences. I worked in retail, banking and all. We don't want to understand too much. We just want to predict whether he's going to buy the credit card, whether this guy is going to buy baby products or not. So understandability, we give importance not to the extent. Not to the extent, but in life sciences, explainability is very important. That's where regulatory authorities will be behind all of you. At the same time, the interaction effects, Professor Goop explained the interaction effects, the non-linear effects. Better you understand before going at finding out the optimized factor settings or predicting whatever your efficacy, dissolution, all those responses, statistically speaking, and your factors and responses. Thank you so much. Good question.
Thank you, Dr. Murli. There are some specific questions as well. I think I'll again pass it on to Dr. Vandana to just contemplate, and we'll ponder over other questions to other speakers. One young researcher has asked, is there any relevance of applying QBD on wearable devices or biosensors? Similarly, how microchips or microneedles can get optimized, so little bit, on the same, while you, conceive the problem and, contemplate on the possible answers. Let's take one more question. Which one is better, Minitab or Design Expert? Such questions are invariably asked from me, if we take user-friendly as the major factor. Wow, user-friendly-ness as the major factor. Now, you are not going to answer. I'm going to answer, because you, Dr. Muralidhara, if he asks, it will be biased, In fact, I personally feel both are good, but Design Expert from DOE point of view is more explicit. Dr. L.D. Patel.
Yes, sir. It is more friendly.
Yeah,
it is more friendly.
But it serves only DOE purpose. Minitab is a general. Minitab is a general purpose statistical package which serves many other needs. And is much more affordable from cost point of view as well. Much more affordable. So there are advantages, disadvantages. Each software has got its own pros and limitations. You have to look into which suits your need. Then, do we apply QBD in every experiment from start to finish of product? Dr. Jasmine explained it, but please elaborate a bit more. She says that QBD is to be applied from start to finish? Yes. Dr. Vijay Sheet Sagar may be able, because he's from industry.
Yeah, I think it is not important whether it is start or middle or end. We are first trying to identify what are the critical processes.
Yes, processes.
And once we identify those critical processes, all of them could be starting processes or middle processes or the processes which are penultimate processes. So that is where we are going to pay our attention, and of course applying the Pareto's principle that whatever makes maximum impact on the quality and the quantity of the product, those factors will be considered first and those processes will be considered first. So I think you should not bother whether it is a startup process or the middle process. As long as it is a critical process, everything has to be addressed in the queue.
In a way, let me, answer in a different way. Your take, I'm retranslating that. Of course, it starts from the start till end, but in the start, you have to conceive that what we are going to be, targeting at QTPB, and what are the CQAs, what are the CPPs, what are the CMAs. You start with right from the very beginning. But as far as the question is being confused with DOE and QBD. DOE is carried out in product development while prioritizing and while optimizing various parameters. Well, the process, as Dr. Vijay Sheet Sagar ji has very rightly pointed out, has to start with the criticality of the input and the output factors in the mind. So I think Dr. L.D. Patel has something down his, tangible things down his sleeves. You can add.
So at every process, QBD can be applied in short, in one sentence. Yes, Vijay sir.
Yeah. But we will will give our stress on that, provided it is making a significant impact. If it is not making a significant impact, we don't have to burn too much of midnight oil on that, so that otherwise, our work will go up too much.
Yes, right. You are right, sir.
Sir, Dr. Vijay Sheet Sagar, couple of small questions for you to better answer. Although any one of us can answer, but you are the most effective player, I think. Difference between PAT and QBD implementation, number one. Number two, what is the purpose of endpoint testing during QBD? Endpoint testing.
First, PAT and QBD. I think PAT is a facilitator for QBD. So, since you want to have a lot of experiments done in a automated or semi-automated fashion, you need to have PAT tools in place. So, without PAT, many times it is difficult to apply the effective tools for the QBD. Very simple thing. Many times I ask the people that, do you have PAT in your industry? And many times the answer, no, we are small, we are medium scale industry, and then we don't have PAT tools. So then I ask them, do you have online pH meter? They say yes. Do you have online conductivity meter? They say yes. Do you have a software which picks up the temperature RH of all your stability chambers on a continual basis? They say yes, but still their answer is no when they ask, do you have the PAT? So I think that basic understanding, what is to be considered as a PAT, wherever you are able to get a continual or continuous output measurement of the process, that's the PAT. So you need to distinguish PAT. PAT is just a facilitator for QBD. PAT is not a technique per se. This is just facilitating what you are doing in QBD. So if you have a lot of PAT tools with you, you'll be able to do experiments in a much more rugged and robust manner. And if you don't have the required tools, then you will not be able to do multivariate experiments. You will be working in a traditional fashion. So that's what I would like to add here. Thank you.
Thank you, Dr. Thank you.
Yeah.
Next is changing within the control space
There was
requires.
one more question, subsidy question, which you had spelled out earlier.
Yeah, and testing. And testing.
Endpoint testing.
Yeah, endpoint testing. So what was the exact, can you read the question again?
Yeah. What is the purpose of endpoint testing during QBD?
I don't know whether we have heard about RTRT, real-time release testing.
Yes, sir.
That if we have a implemented QBD in process, if you have a lot of assurance which is generated out of your overall process, you don't need to even test your product because so much of data is generated, so much of measurement is done during the process. So no need of testing. So you have a route called real-time release testing, which is an official now guideline. So you have to go to the regulator and seek the permission that, okay, I don't want to test my final product. I want to follow it as a means to generate my inbuilt quality, not the end quality. Not end quality is not important because I'm having an assurance that everything is done properly, so the product will be good. If the regulators are convinced, they do give permission to release your final product without doing the testing. So we need to understand that QBD came into existence basically because earlier we were concentrating only on the endpoint testing or the end product testing and giving the certificate that this is within the stipulated pharmacopoeial limits. That was the endpoint. We are no time bothered about the consistency of the process as long as it is assays within 95 to 105%. But if the one batch is 96%, another batch is 104%, we are not really bothered at that point of time. And this was the point I may like to raise, that in one of the forums where US regulators, European regulators were there, I raised this issue to them that what are we talking about the bioequivalence today? Are we really adding any science to that? Because I do everything taking one batch of the innovator and I try to prove that my batch is okay. But I know very well that if I select another batch of the innovator, my product is not going to be okay. So to whom are we fooling actually? Means, are we really adding science into what we call it bioequivalence constantly? So innovator has done a particular methodology. Based on that, he manufactures the product. There is no assurance that his batch to batch consistency will be there. And that is basically that QBD came into existence. And now because of that, we have fair amount of consistency in batch to batch output of the innovator. So now whatever bioequivalence we do carries much more meaning than what was being done in past. So that's what I would like to add. Thank you.
Thank you, Dr. Vijay Sagarji. Now, two questions with respect to control and design spaces. One question is how to derive control space precisely, control space, and quantitatively in design space. Very ticklish, Sara QBD ही पूछ लिया, है ना? Or just we have to randomly select central part of the design space. No, control design space have regulatory importance. Control space, I try to, sum up in my presentation as well. You'll try to squeeze, जैसे you have, 1 lakh, as per mensum as your salary. And, initially you used to spend the entire amount, on the daily needs. But and during the course of, spending, you realize that I have to save something for my future or for buying a, flat of my own or car of my own. Then you slowly and slowly, got an idea that I can manage within 60,000, 40,000. I can every month I can save. So it is just like that. You had these space of enormity of flexibility of spending 1 lakh, but you could manage well within 60,000. So design space is available to you, which is which has got regulatory approval, but within that you tend to remain within the control space, so that any time you go beyond, uh, the safety measure is there. So it's not always the central part, you know, of the design space, which is the control space. I think, you know, the stalwarts, Dr. Vijay Shirsagar and Dr. LD Patel ji can add to it. But another question is, changing within the control space requires regulatory approval, yes or not? Very rightly, the candidate has asked, you know, ये बस मुझे इतना बता दीजिए कि yes or no. No, regulatory approval control space के लिए नहीं चाहिए. ये आपके घर की बात है. ये आपके घर की बात है. डिजाइन स्पेस के लिए अगर आप चेंज करते हैं तो आपको फाइल दोबारा करनी पड़ेगी. You know, it's a new system, है ना. It's a new product, है ना.
So एक QBD का बड़ा एडवांटेज रहा है कि इसके पहले हमको छोटे छोटे चीज के लिए वेरिएशन फाइल करना पड़ता है. But because of QBD, I have proved that within this much of design space, if I have a temperature between 40 to 70 degrees centigrade, if I follow any of the temperatures in between this, the quality of my product is okay. Earlier, if we had fixed it to say 50 plus minus 5, and if you are moving to 45 or 40, you have to file the variation and get the permission from the regulator. But today, the big advantage is that moving within the design space is not requiring any regulatory approval. So it is already a proven design space. So you don't have to do anything extra.
Proven acceptable range. You know,
proven acceptable range. So as long as I'm operating within that, then it's fine. Okay, there are people who have now further gone to near operating range and all that. So they will like to further restrict themselves. That's good science. But coming to regulatory approvals, QBD is going to help us a lot in minimizing our occasions where we have to seek the regulatory permissions. So that's what in a nutshell I think.
Yeah, Dr. Patel, do you wish to add?
Yes, sir. Yes, I will add, sir. In reference to Six Sigma, we explain to the students what is control and what is design. Generally, in Six Sigma, sir, specification, product specification. Are you hearing me? My voice is okay, sir?
Yeah, yes. Go ahead.
Specification are fixed by industry or by formulator as per the requirement of the customer. So that is the design. But when we produce it, when we prepare it, we fix specification of machine that it will give this much variation. Suppose I want to prepare 100 milligram of tablet. So my expectation is 100 milligram, but machine will not give exactly 100. It may be 96, it may be 102. So that is controlled by the technology. So in Six Sigma, we design in that way, sir.
Yeah. So and let me pass on to Dr. Vandana ji for delving on the two questions, you know, which were asked in relation to drug delivery, you know, initially. Can you throw some light?
Again, majorly, I feel that there is a confusion of participant between DOE and QBD. So QBD principles almost remain the same. One needs to actually identify by which process they would be making. So process comes first, understand the material really well, which you are going to use, and then correlate the critical material attributes and process parameters to your critical quality attributes, which are again predefined. So as far as, drug devices are concerned, I had very close experience working on stents, basically, which are coronary stents. And these stents, we were coating nanoparticulate systems to form drug-eluting stents. And these are wearable devices. They stay in the body for a longer period of time. So in this case, again, we had to list because they are going to remain in the body for and then become part of body eventually. We had to list quality target product profile really very well. Remember that these will be treated similar to parenteral dosage form. So all the target profile which is for a parenteral product has to be justifiable here too. As far as material characteristics were concerned, since we were talking about cytotoxic drugs being used, because our major aim in case of stents is that they do not allow the smooth muscle cells to proliferate too much, because if they proliferate too much, again the artery would be sort of closed. So once you have opened the lumen, that should remain open. So in that case, material attributes with respect to this drug characteristics were important, and many of the drugs which are being used here as cell growth inhibitors again exhibit different crystal forms. So it was very, very important for us to look at that particular attribute very critically. Talking about the process is actually similar. You'll be surprised that I had given a paper design to that company and they fabricated a machine. So if you understand the coating process well, these are spring-like structures which need to be coated. So again, it has to be rotated while it is being sprayed. So in that case, again, what was critical to us, we found some trial and error batches, of course, were pre-run, but we found that it was majorly the spray rate. And since they are nanoparticle based, we had used some sonication energy too, so that they remain in the nano form after coating too. So spray rate, the sonication frequency, and inlet and outlet temperature played a very, very important role. So once we understand this part and we know which are the basic parameters, all we had to do is apply DOE, which in this case was box Benkem. We went forward and made stents. I think this has given you a fair idea as to what are different steps. Again, coming to understand your process material very, very well, and then any product you can design, I feel, be it any kind of transdermal variable devices or it could be internal devices like stents.
Thank you so much, Professor Vandana Putravalaji. Let me share that Professor Vandana has more than a score of, I do not have her CV with me, score of patents and almost, nearly 10 technology transfer. And one of the recent technologies which he has transferred is drug-eluting stents of coronary, for coronary artery disease, Fantastic. There could not be, a better person than you to elaborate on this aspect.
I think we also strongly noted that there are many researchers, but that research has to come into the commerce. So she has been very successful in commercializing the products. So that research has really benefited the customers. I think that's the good. Otherwise, many people do research for the sake of research, for PhD and all that. So, but I have seen some products successfully rolled out.
Yes. Yeah. So I told
in some my dear parents, I suppose.
Really sir, so at so many places I told that she is researcher academician.
Yeah.
Researcher academician.
Researcher academician. And certainly, commendable. Kudos to you, Doctor. And then there are questions like, I think more on statistics. Anyone familiar with the quality of multiple linear regression analysis model for CCD, R-square of prediction? I think I pass it on. I can elaborate this myself, or Dr. L.D. Patel can also elaborate, but will prefer it to hear from our young colleague, Dr. Muralidhara.
Yes.
And
JMP, yes.
Before that, Dr.
You want to jump to Dr. Murli?
Yes.
Doctor, what is adjusted R-square and F value? Not only the significant value, but what does, what is the significance of that? Adjusted R-square. that's the R squared adjusted for degrees of freedom, and F value, I think, which is of tremendous importance. These two questions, you have to answer.
Okay, so I will take the F value. Once we move from simple linear regression to multiple linear regression, again, we are restricting linear regression. Again, we are not getting into the classification, which is another area. Professor has been talking about it, because that's where we have a lot of other metrics, because simple linear regression. So R square, we are looking at whether X value is the variability in your Y value significantly being explained by your variability in X values. It's always look at the average relationship when we talk about simple linear regressions, averages. So in R square and F value is a whole model test, we call it as, whether together all the variables to an extent are influencing or variability is being explained in the Y value. That's what we call it a whole model test. We look for analysis of variance. At R square shows that it's a coefficient of determination. So it varies from 0 to 1. But we always ask this question, what is adjusted R square? R square has a tendency or a limitation. More the number of predictors, more the number of X variables you put, it will automatically improve. Simple as that. I always tell the theory of parsimonious, that's my favorite. How can you find out the so important top 3 or 4 variables so that you can control? As a human beings, we cannot control everything. We don't have to control everything. So we look at those 3 things. We always look at what is that percentage of variability being captured, 70 to 80. In chemistry, 90%. In pharmacy, if you ask me life sciences, it should be higher. Social sciences, if I can able to predict my daughter's behavior even by 40%, I'm more than happy. R square of 0.4 is more than happy for me. That's a proud father talking here. You can understand. So it depends on not only the text, it's always the context. In life sciences, it's always higher the better, but it depends on what is acceptable range to an extent. And the second part is whole model test. If you look at multiple linear regression, each individual variables, you look at the parameter estimates and the significance, 95%. You look at whether it's statistically significant or not. Essentially what you're looking at is, can I generalize this? Is it a representation of my population? Can I make a general statement out of it? That's what you're trying to do it. And if whole model is making sense and you look at adjusted R square just to beat the R square for the number of variables. You don't want R square. By the way, adjusted R square can go negative. That's one of the interview questions I always ask in data science. Hey, can adjusted R square be negative? No, sorry. Adjusted R square can go negative. So you always look out for penalizing for the number of predictors, because higher the number of predictors, you keep pushing the variables, R square get inflated. So you want to penalize for number of, in statistical terms, adjust for the degrees of freedom. So how many independent units? So predictors. So that's the reason you look for adjusted R square. You don't want to overfit a model, right? Overfitting is just saying that 0.90%. Yeah.
Thank you.
That was a long answer for the short question.
I think the carry home message from what Dr. Muralidhara, shared with you, the wisdom which he shared with you, is that adjusted R square as well as the significance of F significance ratio or F ratio, as Dr. L.D. Patel earlier pointed out, the inferential statistics or inductive statistics is based upon what is explainable upon what is unexplainable. Simple, as simple as that. So R square upon 1 minus R square gives you F value, and R square corrected for degrees of freedom gives you adjusted R square. So both tends to, negate the influence of N. R square is dependent upon N value. As N increases, R square tends to increase. So if you tend to take up adjusted R square and F ratio, F value instead, so those, that significance, that influence of N on R square is taken care of. A very interesting question. I think probably, according to me, the most interesting question, which everybody of us can, throw some light on the same. The candidate, out of some, information and some frustration, probably, he has asked, what is the future of individual's intellect? What is the future of individuals' intellect if artificial intelligence, QBD, and softwares like GMP, Minitab, Design Expert take the major chunk credit for the development? No, I think, मैं छोटी सी बात कहूँगा, there is no surrogate to your experience, your knowledge, your know-how, your skills, and your art, because pharmacy is both art and science. So उसका कोई surrogate नहीं है. These things are required. In addition, Dr. Vandana and I were attending to one very informative talk before that by Mr. Sanjay Sinha from IPCA, and he threw a lot of light on the contemporary trends towards continuous manufacturing, artificial intelligence, virtual reality, PAT tools, and of course some QBD elements, and the OEE, that is overall equipment efficiency, etc. These tools are required today to to supplement what is the intellect's ability. I think Dr. Vandana, Dr. Patel, because this is an from academic point of view, I think they can throw some light to encourage, I think, the researchers. Or should I move to the another? How to visualize or conceptualize academic research? I think I'll pass on to Dr. Vandana. How to visualize or conceptualize the academic research? I think it needs one day deliberations on the same and a couple of lectures from Dr. Vandana itself. Till what extent these advancements to be implemented in the academic research? A couple of lines of wisdom by Dr. Vandana.
So I believe, actually, when I started, I didn't know ABCD of QBD. It is only by, let me confess here, Professor Boop, it's only by reading the literature which you have published, we have learned whatever we have learned. Let me be very frank on that. And that grit and determination was there, though Professor Boop had actually offered that you tell us and we will do whatever you need to do, but I wanted to learn it all by myself. And when you have that in yourself, that you want to learn it all by yourself, you tend to read more, you tend to interact with more people, and then in turn, you learn more.
I must appreciate the humility of Professor Vandana Patravale.
Yes.
Rome was not built in a day. Even Taj Mahal was not built in a day. She attends, sir, Dr. Vijay. She attends to the seminars, the talks of every other person who's relevant to the knowledge and the know-how which she aspires to learn further. She's a continuous learner. And that is the point which is currently missing in the younger generations. In majority of the younger generation, we tend to consider that हमने काफी कुछ learn कर लिया. No, जब learning stop हो गई तो earning भी stop हो गई. Earning not in terms of money, and earning in terms of
knowledge,
outcomes. वो भी
बंद हो जाए.
So we have to be continuous learners, continuous student, consistency से हमने studentship रखनी है तभी, because जानना इतना कुछ अनअर्थ पड़ा है ना, तो हमें learn करते रहना, apply करते रहना, learn करते रहना, apply करते रहना. फिर कुछ questions
डॉक्टर साहब 2:11 हो गए हैं. I hope we can continue.
I think 4-5 मिनट रह गए हैं. Sir, there are other questions. अगर organized Dr. Sharik, how far we can stretch this? Dr. Sharik, the organizers.
Hello.
How far we can extend this? 15 minutes?
Yeah, we have time till 2:30, sir. But at 2:30, Dr. Ejaz will be coming in.
Okay.
So he's committed.
So we have 20 minutes, you know?
We have 20 minutes more. Yes, that's it.
Or 20. Okay.
So just sorry to interrupt. I know there are a lot of good questions that are coming on. One question that Dr. Bhoop, I would like you to add to your list before we end. is since there are a lot of young researchers and scientists in this panel, please, I would request all of you to give your couple of lines of wisdom into research trajectories of your lives and your guidance, because life as a researcher, as an academician, is obviously full of roller coasters, rejections, grants rejections, paper rejections. So how have you kept that kind of patience and grit that Dr. Vandana Tafawad was talking about? I think that will be very inspirational to all of us. So one question, I think before we end up, wherever you feel like you should introduce here. Over to you, sir.
Yeah. I think a couple of questions let's address. One question is explain more about of response surface methodology and models like cubic, quaternary, and so on. Let me share. Cubic kind of model is rarely adopted in pharmaceutical practice. Either it is linear or it is quaternary, or another which you are missing is interaction. So invariably, we tend to look into linear interaction and quaternary models, and almost all the software which you use are capable of the same. And particularly for response surface methodology, linear model hardly works, except when you are using designs like full factorial design, or it may sometimes come to your rescue, but normally it is the non-linear, quadratic, and interaction models. Dr. Muralidhara and Dr. L.D. Patel can correct me if I am wrong somewhere.
Absolutely right, Professor.
And then how can we use QBD in formulation and evaluation of FDCs? FDCs, it's a very complex ball game, I think. FD, fixed dose combinations. So उसमें एक ड्रग के लिए ही बहुत मुश्किल हो जाता है, डिफरेंट ड्रग्स के लिए QBD करना. QBD is in fact, as Dr. Vijay Sagar was pointing out initially, is basically drawing the understanding the system. If I keep sharing that if a doctor commits a mistake, it is the life of that patient under peril. But if a industrial scientist commits the mistake, grievous mistake, it is the life and health of hundreds of thousands of the consumers who are consuming his product.
Society, sir.
Yeah, society. So one has to be very cautious. So in fixed dose combinations, using various approaches, like I think Dr. L.D. Patel can throw some light, like chemometry, etc. You can draw some understanding, but as such, if you wish to adopt, DOE for this very purpose in a very myopic manner, I think that will not be the purpose to be served in FDCs. Dr. L.D. Patel, sir, you can throw some light.
In fixed dose combinations, sir, we have to decide so many factors. First of all, not only single factors or single parameters, and again there may be interaction also.
Yeah.
So it is not a linear
API interactions. Yeah.
It is not a linear one. It is a quadratic or complex.
Yeah.
In that way. So, and there may not be two parameters. There may be multiple parameters. So simple factorial design may not be well in initial. At the end point, it can be applied. In the initial, we can have BB design. So when there are so many factors, out of that we have to pick up 3, 4, 5, and one of the response, and then again we have to do factorial design. I think that will be the proper method.
Thank you, Doctor.
I'm okay, sir. I'm okay.
Take it, Doctor.
Yes.
The candidate should be more
satisfied.
Okay. So, Doctor, one question direct to Dr. Vijay Sagar. How is QBD approach involved in method development during in vitro dissolution studies? I think you have been talking. You are the most proficient,
Dissolution
expert in
master.
this area.
I hope the person who asked the question could attend my presentation. But just to narrate it in brief, QBD approach is extremely important for dissolution method development. It starts right from the condition which can give you the best deaeration. That's what I told you, that whatever medium we use in dissolution needs to be deaerated in most of the cases. So we want to have the minimal content of dissolved oxygen in that. So you have the parameters to play around, like temperature, vacuum, and the time for which you will expose it to the vacuum. So what is the best condition that can give you the minimum amount of dissolved oxygen? It starts right from that. Then we have so many apparatus choices today. You have got 7 apparatus. Now the peak apparatus is also getting added very soon. So you'll have a battery of 8 apparatus. Which is the best apparatus for my product, and which is going to give me a discriminatory dissolution method? That's what I'm going to concentrate quite a lot there. So apart from this deaeration aspect, we have the parameters like we have the particle size, we have the dissolution time for which you are going to dissolve the product, you have the RPM that you are going to use there. So there are at least 3 factors which make a very significant impact on the dissolution of the product. So can I do the experiments there the way I suggested in my presentation and try to find out what is the best condition to get the optimum dissolution? Now, whether that optimum dissolution is discriminatory or not, that's a second dimension. So first I'm trying to see that in what condition I'm going to get the optimum dissolution of the product. And then I'm going to get into my, say, pilot bioequivalence studies and get some PK characteristics on a product and try to compare which method is giving me the picture which is mimicking what happens in vitro. So if in nutshell I have to answer your question, extremely important to apply QBD for dissolution method because it has to be discriminatory. And if it is not discriminatory, whatever efforts you took on the development of the products, where you have spent so much of energy, so much of time, all will go in dust. And as I was explaining in my presentation, even today, the failure rate of the products failing in bioequivalence studies at the final stage is pretty high. So it has come down over a period of time.
And the major reason is dissolution.
Yeah, and
because
that's the reason why we formed a society called Society for Pharmaceutical Dissolution Science.
Because most drugs tend to be either BCS class 2 or BCS class 4 drugs.
And I may like to tell the audience that Dr. Bhoop, Dr. Vandana, all are contributing in their own way towards the development of this society called Society for Pharmaceutical Dissolution Science.
And they are organizing, society is organizing DRPI. And I'll, on behalf of Dr. Vijay Sagar, who has been the president of SPDS for quite a number of years. So on behalf of all, I'll share the, brochure with Dr. Sharik, and he should.
Dr. Bhoop, during my presentation, I already sought the permission of Dr. Sharik. And at the end of my presentation, I briefed for about 2-3 minutes on this DRPI event. So all of them are.
It is 31st. I think they can still submit the abstracts if they wish.
And it's very genuine. My student last year scored 40,000, second prize, and bagged 40,000, and she floated a party. I can vouch that.
I showed even their photographs also.
Good, very good.
Sir, very, very interesting questions, 2 questions, which can be clubbed together. How to design a chemical moiety with the help of QBD, number 1. Number 2, how is drug structure characterized can one apply manual standard deviation values in QBD tables? I think there is little confusion with respect to the second, but with respect to first, it's very well taken. The approaches like multiple linear regression analysis, principal component analysis, partial least squares, discriminant analysis, cluster analysis. Dr. L.D. Patel, is an expert in that area.
Sir, you are more expert in that way as compared to me.
Sir, I have very high regards for you for your expertise. So these approaches can be used,
Yes.
what's called as combinatorial, part of the combinatorial approach for designing newer chemical entities. The software which are being used are also banking upon these techniques, multiple. That's chemometric approaches here. Dr. Patel can highlight some. Dr. Patel, sir. And with respect to the how is drug structure characterized, can QBD help? I don't think. Structural elucidation, QBD role उसमें है ना, वो तो NMR, mass or CHNOS analysis.
Docking and other things may be used there, not QBD, sir.
Yeah.
There are some chemical softwares, chemistry softwares should be used.
Yeah.
Not DOE or not this one.
Now, as Dr. Sharik advised, a little bit about your research sojourns. I'll request Dr. Vandana Patravale, probably the most accomplished researcher amongst us, an academic researcher. So I think, and academic researcher with, industrial overtones, with industrial outcomes. So I think let's start with Dr. Vandana Patravale. As far as research is concerned.
Very, very high regards for both of you, Professor Patel as well as Professor Bhoop. Yes, as I said, learning has been from our senior professors. As far as research is concerned, Dr. Sharik, I divide it into two parts. On one side, I keep my academic interest always alive. On other side, I tend to cater to needs of industry. So there are two kinds of projects which we do undertake in our lab. One, which comes from basic sciences and then reaches applied, and it may or may not reach industry because their industry is very slow. But as far as the other part is concerned, yes, identifying what industry needs and working towards it. So my modus operandi always has been to look at where there are unmet challenges, identify them, and I believe that in India, majorly, why I'm working a lot in infectious diseases, that is the reason. People are working on lifestyle diseases a lot, but our country as well as some other developing countries are faced with innumerable challenges which no one in the world is even looking at. So like neglected diseases. So my major focus always has been that. And since I'm a formulation scientist, I believe that whatever reaches patients has to be definitely very safe and efficacious. So I have focused more or less on lipid-based drug delivery system because all of us are taking lipids in some form or the other, even in our food. So I believe if food is safe and we are surviving for so long, definitely my delivery systems will be safer too. So with that conviction, we are moving ahead. You also asked about, did we face any challenges? So yes, like any other beginners, we did face challenges. And I remember I was, say, maybe 5 years in service. I joined ICTE as a lecturer in 1991. Before that, I was doing my master's and PhD also there, which was UDCity then. So I went to my HOD and started crying. He said, what happened? I said, I have sent my publications and I've written them myself. It's not that students have written, I've written it myself. And still the publications are not getting accepted. It's coming back from the editor. He said, don't worry, I took 15 years. So that actually built my confidence so much that I didn't give up. But meaning there were times when, you do go low, but there have to be someone who can do hand holding. And that is why, you know, I am not saying no at any forum. I'm trying to encourage all the youngsters that don't give up. Please, there people like us are there, you know, Professor Bhu, Professor Patel, Professor Shirsagar. I mean, they are such senior people and still coming to your level and talking to you. Why? Because we want, this is a nation building exercise. It is not personal exercise. Basically, we want our nation to prosper. And if you keep that in front of you, that motto in front of you, that whatever inputs I am giving, it is my India which is going to benefit, I think nothing can come in your way.
Yeah.
So with that in mind, we are moving ahead. As far as research grants are concerned, now sort of we have mastered the art, I would say. So my success rate has been quite good, I would say. It is 65%. So definitely now we have mastered the art, but then at various forums, I have given lectures how to write a research grant. What is it which, you know, government authorities are looking at when you are writing a, and I would put it in this forum that please go through high risk, high award SERP grant. Why I'm saying that? Because they in bracket have explained what is expected. So if you understand that grant well, you can write any grant. So go to high risk, high reward grant of SERP, and I'm sure you'll get all the answers. I suppose, Professor Bhoop, if you want anything else, I can definitely add.
Yeah,
but
good enough. Thank you so much. Thank you so much. Dr. L.D. Patel ji.
Thanks, sir. Actually, I'm a teacher, and I started the research from UG level. We given LipGuard, sir. You may have heard the name LipGuard. LipGuard is our LMCP BPharm student products. We given experiment set-wise to the UG students.
100 pages.
4th year dosage form design and farm technology students. And we formulated that LipGuard. And that formulation was given to that pharma company.
Wow, great.
And that is, sir, that is a story of 1989.
Wow, great, great.
And Dr. M.C. Goel, late Dr. M.C. Goel, is the nucleus around this. We were young and fresh at that time. I joined in 77. I did my master in 77 from LM College of Pharmacy. So we started. So we started using UG students, and Ahmedabad is hub of pharma, and you know that there are so many small scale companies. We take their formulation, what product they are preparing. We take that high earning formulations voluntarily with all compositions, and we give them guarantee that we will reduce your cost at least from 1 paisa to 25 paisa using your principle, that is DOE principle, sir. And that work was done through PG students partially.
Great, great.
We have to also work. Some trials, some important trials we have to also take. And there such voluntary consultations, I had did more than 250 formulation cost optimization. 250.
Wow, great. And all voluntary, complimentary.
They give us TADA only, sir. Okay. We demand only TADA, nothing else. Why? Because
service, industrial service. Wow.
Yes, no, sir. Why? Because they are our students. They are my LMCP students. So we take their one product and learn them how to optimize their things. So that is my research funda. And today also we are in contact with industry. Within last 1 year, we prepared so many Ayurveda formulations.
At Parul Institute?
Just now I'm at Parul, but I was at LMCP up to 2004. Then I shifted to Baroda. My son is in Sun Pharma. He's BPharm, MPharm, PhD, and he's in Sun Pharma. So I shifted there, and now he's transferred to Ahmedabad. So we have base of Ahmedabad and Baroda pharma companies. Everyone may know by our name, LD Patel. They may haven't seen us, but they may know.
Yeah.
So that is our asset, sir.
Great.
I want to give advice to our participants that daily we have to read at least for 2 to 3 hours.
Yeah.
Today also, I'm reading or I'm attending these webinars and delivering webinars also. So we have to read 2 to 3 hours daily. We have to update
continuous
that.
learning. Yeah.
Continuous learning and updating it.
Yeah.
Without that, nothing can be changed. That is one thing. Second things, our approach should be polite and polished. Our approach should be polite and polished.
Oh, great.
Nothing can be demanded. Everything can be achieved by your efforts.
Yeah.
Okay, thank you, sir.
Thank you so much, Dr. L.D. Patel. I would request now Dr. Vijay Sagar. You know, whenever I hear Dr. Vijay, I see a teacher in him. Although he has every inch, he has been a very successful industry scientist and a leader. I have seen his leadership at SPDS as well. So I have been the North Zone president. He has been the president overall of the country SPDS. So I have seen his organizational abilities and his leadership, and he's a highly sought after consultant in industry. After leaving the active work. So by every standard, you are an academician too, sir. So please share your.
Thank you so much, Dr. Gu, for your kind words. As you know, I'm mainly from the industry and having spent more than 4 decades in the industry. What is the paradigm shift which has taken place in research? I may like to explain it by giving an example. That I was leading the quality team and we had the product developed by the R&D. And many times when the technology was transferred to the manufacturing site, we used to have problems. And there used to be tug of war, blame war, that you don't know, you can't work properly, everything goes fine in R&D, but your people at the plant level do not know, they don't follow the discipline, they don't follow the SOP, so on and so forth. So when we used to hear this in the meetings, I said, now enough is enough, because I had done my studies. And then I told our MD that, look, according to me, there is something going wrong in the development. And you need to really make it explicit. I would not talk, though I have already understood what is happening, but I would like to have a proof of it and then come back to you. And they said, what do you want to do? So they said, I would like to audit the R&D. Immediately R&D guy said, what business quality has got to, the audit the R&D processes. We are developing the products, we are transferring the technology. So as long as we are transferring successfully, they should not bother much. But then with the consent of the MD, I could break the ice and get into the R&D and saw how they are. This is, I'm talking you quite significant years behind, I think about 20 years behind. So when I said, how did you freeze the technology? They said, we did the 3 experiments. I said, okay, show me the output of those experiments. With the same quantity of the starting material, in one case, the yield was about 35, second experiment was about 45, and the third one about 23 grams. When I checked the quality, one was white, another was off-white, third one was yellowish white. When questioned, the approach said that yes, all of them are within the specification. The description is white to yellowish white. It fits into that. And what you have got to do with the yield, as long as the quality is fine. So that was the research in past. So now you are all young people. Now you have to understand that today we have to do the research for the sake of benefiting the end customers and benefiting your company from the cost and quality point of view. So as long if you keep that in your mind, if you get focused onto that, understand the consistency out of the output, I think you would be the best researcher, you would be the best contributor in your industry where you are. Thank you so much.
Thank you. Thank you so much, Dr. Vijayji. Now, I think we are running short of time. I'll take a couple of minutes in sharing my sojourn.
Sure.
Regarding...
I think Dr. Ijaz has not yet joined, so we have the liberty
Dr. Ijaz?
Dr. Ijaz has not yet joined, so I think we can have that liberty of extended time, I guess.
Yeah, thank you so much, Dr. Shri. So, मैं सारी लाइफ की रिसर्च सोजॉर्न ना टच अपॉन करता हुआ, but which is marked with so many vicissitudes, ups and downs, you know, in my professional life, but never, as Dr. Vandana shared, never ever deterred, kept on, you know, moving as per the holy Gita's philosophy. कर्म है हमारे हाथ में, फल नहीं है. फल उसने कब देना है, कैसे देना है, वो लॉर्ड जाने, है ना. हमने तो अपना कर्म करना है, लॉर्ड है ना. सो वो उनको पता है ना, लॉर्ड को पता है और देता है. तो कई बार मच मोर देन योर एंटिसिपेशन, मच मोर देन योर एक्सपेक्टेशन देता है, इतना छप्पड़ पाट के देता है. सो मैं एक छोटा सा 1992 टू 1994 मेरे दिमाग में एक इंट्यूटिव आइडिया आता था. आई थिंक डॉक्टर मुरलीधरा should pay heed to it, that you know, in all the pharma industries whichever I have visited and I come across, the development, formulation development is not being done in a satisfactory manner. मेरे दिमाग में होता है यार कुछ तो गड़बड़ है, जैसे वो CID की मूवी में आता है ना, कुछ तो गड़बड़ है ना, कुछ तो है, गड़बड़ है, नहीं है, ठीक नहीं है. Then I got one of my very good student, जो गेट होता था उसको, तो गेट टॉपर था वो लड़का. I would give tributes to his name, Raghavendra Kanti Gupta, Raghav Gupta. Now he's in USA. So he, you know, I found him very intelligent. मैंने उसको फार्माकोकाइनेटिक प्रॉब्लम दी हुई थी M.Pharm में. तो मैंने उसको कहा कि यार राघव, छोड़ उस फार्माकोकाइनेटिक प्रॉब्लम को, मेरे दिमाग का ये कीड़ा है, इसको सेटिएट करना है. बड़ी देर से मुझे तंग कर रहा है कि कुछ, you know, कुछ सिस्टमेटिक होना चाहिए, कुछ बढ़िया तरीके से होना चाहिए. इस तरीके से नहीं कि यार ये फॉर्मुलेशन अच्छी लगती है तो बना दें, है ना. यू नो, वन फैक्टर एट वन टाइम, जैसी अप्रोच मुझे ज़्यादा नहीं पता था, पर मुझे इंट्यूटिव फीलिंग, गट फीलिंग के साथ मैंने उसे शेयर किया. उसका कीवर्ड्स क्या है, मैं कोई कीवर्ड्स नहीं, सिर्फ ये है कि वो सिस्टमेटिक होनी चाहिए और कुछ प्रोग्रामिंग होनी चाहिए, कुछ उसमें पता करें, है ना. सो आई गिव यू 1 वीक, मैंने उसको कुछ बता दिया. ही गॉट द जिस्ट. सो 1 वीक के बाद उसने कहा नेक्स्ट मंडे आई आस्क्ड हिम टू रिपोर्ट. उसका कहना सर मुझे 1 हफ्ता और दे दीजिए, नेक्स्ट मंडे, उससे नेक्स्ट मंडे आई रिपोर्ट. एंड देन ही ब्रॉट अराउंड 30-40 यू नो पब्लिकेशंस. यू नो, उसने कहा कि इसको सर जो आप कह रहे थे ना, इसको ऑप्टिमाइजेशन, सिस्टमेटिक ऑप्टिमाइजेशन, डिजाइन ऑफ एक्सपेरिमेंट्स, ये सब कहते हैं. मुझे, you know, we didn't know, we never studied Muralidhara, never studied, you know. So मैंने कहा कि यार ये मैं पढ़ता हूँ. हमने एक आध हफ्ता उसको क्रिटिकली एनालाइज किया और डिस्कस किया, है ना, 1-to-1, एंड वेरी पैशनेटली, वेरी फर्वेंटली, वेरी एंथुजियास्टिकली, वेरी जीलसली कि हमने कुछ सॉल्व करना है. फिर मैंने देखा कि यार सॉफ्टवेयर चाहिए डॉक्टर मुरली, यू नो, सॉफ्टवेयर चाहिए. सॉफ्टवेयर वी, you know, सॉफ्टवेयर हमारे पास था नहीं. आई एम टॉकिंग अबाउट 1995 आ गया, 1994 में शुरू किया, 95 आ गया, है ना. दैट्स अराउंड 26 ईयर्स बैक. कोई सॉफ्टवेयर नहीं. हमने बड़ी बड़ी जगह लिखा, कमर्शियल तो था ही कोई नहीं खास हमारे पास. हमने लिखा तो हमें, यू नो, कोई पॉजिटिव रिस्पॉन्स नहीं आया. एंड बाय दैट टाइम आई हैड डेवलप्ड 12 सॉफ्टवेयर ऑफ माई ओन, बिकॉज आई लर्न Fortran, आई लर्न C++, एंड मैंने अपने सॉफ्टवेयर लिख चुका था, खास करके जोरल, जीरो ऑर्डर लीड्स वाला, तो बहुत यूजफुल एंड पॉपुलर हुआ. तो मैंने कहा फिर यार हमने 4 महीने इसकी रिक्वेस्ट में लगा दिया, कर तो हमारे पास फंडिंग तो है नहीं इस वक्त, है ना, तो हम अपना डेवलप करते हैं राघव। वो कहते सर मुझे आता नहीं, मैंने उसको I trained, got him trained, and we worked together from morn till eve. So within almost 6 to 8 weeks, we developed that software factorial optimization. And subsequently, we added the components of central composite design and mixture design into it. And we solved, our purpose. I still remember at 11:45 p.m. we both were working in the department when we could generate 3D graphs, using the software. And हमने बगड़ा डाला था वहाँ पे. So to cut the long story short, we worked on using that software. 6 साल हमने काम किया till we found that we are underprivileged because the software which was available to us, that's design expert with somebody and Minitab. So वो और बढ़िया करता है. Then by that time I had a project, so I bought a design expert worth 45,000, and we started working 6-7 years. So we started working with our software Factop. We have almost 7-8 publications out of Factop alone. So where there is will, there is way. आज मेरे पास 410 के करीब पब्लिकेशंस है मेरे ग्रुप, मेरा नहीं कहता, मेरे ग्रुप के पास. So 410 में से almost 60%, 55 to 60% are related to QBD DOE in one way or another. This was only due to the persistent and consistent efforts of an MPharm student who danced to the tunes of the guide, and the synergy brought us to the way to the place where we are. So there should not be any difference. Will रहनी चाहिए. Where there is will, there is way. Thank you very much. Thank you. So I would request now Dr. Muralidhara to share, although he's too young, so couple of minutes, couple of minutes.
Yeah, so I was just remembering the word Pushpamala Prasangena Sutram Shirsidharyat. Even an average person like me, if I'm getting to the right network like you people, so even the grass and the thread goes to heaven. So my taking today is that's what it is. So I want to be in this group, and this is the flowers, and I want to remain as a grass and thread. And yeah, so this is what that's what 2 words from my side. I've just been learning. I always say that it's been 22 years working in industry, and I continue to work in industry. I always have a different motives also when I speak, but this is something which I always enjoy, being part of training or kind of these kind of things where I get to learn a lot of things. But always my professors taught me when it comes to statistical thinking, I should say 2 words: be curious. Curiosity creates capability. That's the famous statements. Curiosity creates capability. And learn to earn. Professor said you should earn. Okay, learn to earn respect. Okay, this is what my professor said. So after looking at all of you, you have earned a lot of respect. So you people are inspirational for me. So I'll keep looking up to you people and keep blessing me. Thank you so much for giving me the time. Yeah.
Thank you so much, Moses. Can I ask a question, Dr. Bhoop, if you permit?
Oh, thank you. I'll be grateful. Brother.
So, and this is for all of us, all of the panelists out here, esteemed panelists. A lot of times I see when I move back from US, I think in academia, I moved from industry to academia. It was a cultural shift for me. So I see a lot of young researchers. Sometimes they are pressured into just publications. So sometimes the quality of publications gets dropped, and they're just after just publishing patents, so wo CV bada jana chahiye. Somewhere like impactful work gets lost essentially. Maybe it's just the pressure for them because the promotions requires that. So a credible work, something that gets translated into something rather than just staying into some esoteric papers which probably only the 4 authors read, no one else reads. So what do you think? Because the other thing is challenging. Developing impactful work is challenging. It takes a lot of time. It takes a lot of effort, great perseverance, a lot of rejections there. So what's your advice to young researchers? This is both.
Yeah, I think in this regard, the other 2 experts sitting from academia, they are much more pertinent to answer to address this, if not query, but the point of discussion. Dr. Vandana Patravale has so many tech transfers to her credit. Dr. L.D. Patel has so many complementary tech transfers as he has here. So I do have 3 tech transfers, but all in collaboration with Professor Katare and Dr. Gurpal. So, somehow that commercial sense was poorly built in my psyche. That's why probably that's the reason. But I did use that. I did build that commercial sense while providing consultancy to industry, because that's what I was taught by some of my peers. So I charged from industry when I devoted a lot of time for training them on QBD, on biowaivers, on IVAVC, etc. So I initially I used to do it the greatest, as Dr. L.D. Patel sir told. So but Dr. Vandana Patravale and Dr. L.D. Patel, certainly I must agree with you that publish, patent, or perish. This is the paradigm which should be percolated amongst the researchers. Publish, patent, or paradigm, and publication should not be for the sake of it, for propagation of the science. And patents, wherever that is rewardable, that is, as Dr. Vandana told, part of my research is for the science benefit, and part of it is for industrial benefit, for the meeting the unmet needs. So the second part, part of the research should be patentable and should be patented, and then and the tech transfer should be explored. That's what Dr. Vandana and Dr. Patel have done it successfully in our department too. It started with Dr. Katare. Now everybody is doing. Everybody, be it Dr. Indupal Kaur, be it Dr. Gurpal, be it Vandana, be it, everybody is doing. It has started the chain reaction. So here, but country across, I see Dr. Vandana Patravale as one of the leaders, and I must, ask Dr. Vandana to share some pearls of wisdom, and Dr. Patel.
So Dr. Sharik, I feel that we as academicians, in fact, do not understand our strengths. So first thing is we need to introspect and understand our strengths. Second is where we fail is we have some kind of fear. Why do we feel inferiority complex? I really do not know. If you have understood the problem well and tackled it well, what is stopping you from reaching to industry and telling them this is what I have to offer? So basically, that fear will come if you have not paid 100% attention when the researcher is developing it. I think that is a connect which we need to build with our student and understand at every stage as to what they are doing, so that we can then boast on networking. That is where we lack. I think we need to build it, and you'll be really glad that not only opportunities exist in India, we have been, in fact, catering to many of the products of UK, Ireland, etc. And why is that happening? It didn't happen overnight though. So yes, it did start with publications. People read our publications, understood where we were, how systematically we were doing research. Then came the patents. Patents come in the public domain, so they know what we are working on. And then, of course, the word of mouth and my reaching out to them. I would say my, because at that time, this institute innovation policy was not existing. So it was left to people, to tap the industry on their own. So I reached out, I made brochures, simple brochures, telling them what I have to offer and mailed them to n number of industry people. We need to keep in mind, we need to approach right kind of people. So if teachers think that they know someone in that industry, in R&D, they can help, answer is no. Most of the time, the business decisions are taken by BD people, business directors, and marketing people. So we need to approach them and offer our technology, whatever we have, to them. And I think in that we have been successful, and therefore the returns really have been good. And in fact, I would tell you, first you give something, then you can get anything, whatever you desire. I think Professor Patel and Professor Bhoop, both of them have said that. When I started with the stent company, I didn't ask for name in the patent or getting royalty or anything, because for me at that time, 1997 I'm talking about, it was a challenging thing to take up that project. But now you will be very glad, in fact, to know that just yesterday I've been inducted on their board of directors as independent director. So automatically things are given. I feel
congratulations.
Thank you so much. I thought I must mention this, that in the beginning, don't ask. Typically teachers have that, if they have some technology to give, they'll put a hefty price for it. It is not. Work with giving attitude, and finally you'll be just getting it. I think that answer. Thank you.
Very nice, very nice.
Yes, very nice.
Very aptly said, madam. I think this
Dr. Patel, sir. Dr. Patel, sir.
Congratulations, Vandaben. Congratulations to be independent director of the Sejanand Medical Devices. Congratulations, many congratulations. You are deserving that.
Thank you so much, sir.
Sir,
which medical company? Which medical device?
Sejanand stent making medical devices.
Okay.
They are based in Surat, but they have one of the finest, setups. One should visit it. It's a best setup. By now, there are about 4.5 lakh stents which have been implanted.
Wow.
Yeah. So
just commendable. Let's have 3 cheers for Dr. Vandana.
Thank you.
4.5 lakh stents have been implanted, and the technology was, developed and transferred by none other than President Vandana. Commendable, really, and he's an academic.
Yes.
Wonderful. Kudos to you. Kudos. Salutes, madam. Dr. L.D. Patel, sir.
So congratulations to the researcher academicians.
Yeah.
That is my words. Sir, nowadays, what are the lacuna with the present generation? So I want to advise them some 3-4 sentences. Keep contacts of your well-wisher and your teacher.
Keep contacts
of well-wisher. Well-wisher and your teacher. Why? Because they know you from bottom to the top. That is one thing. Second thing, respect your institute. Generally, people take qualifications, they get degree, diploma, masters and PhD, and then forget the institutes. So give due respect to this institute.
Institute, as well as, let me add, as well as the mentors.
Yes, mentor means guide or teacher.
Very good.
Guide or teacher.
It is in their benefit, not the benefit of
definitely,
the mentor.
Definitely. And if you or anyone feel lack of resources in terms of equipments, in terms of finance, in terms of guidance, keep contact of the good persons, and from that you can get that. Other things, Vandana Ben has told everything, so I'm not repeating that things, but these are the new things which I want to transfer to the participants.
So nice.
Thank you, sir. Thank you, sir.
Thank you so much. Adding from where Dr. L.D. Patel sir left, that is, you have to be connected with your alma mater and also with your mentors. I personally feel, I have observed in me, during our student days, we were very, very respectful to our teachers. We had that awe, that, respect come fear. But we people, I think Dr. L.D. Patel, Dr. Vandana, would agree with me. We people are much more friendlier, than our seniors, our teachers somehow. So, but that friendliness should be respected, should be appreciated, and that blessings should be sought of your mentors. Somehow, in the North India, I observe this to still prevailing in Middle India, like Sagar, like Gujarat, like Mumbai, and but somehow I observe that trend decreasing in Delhi upwards region.
Yes, sir.
जितना रिस्पेक्ट करेंगे, जब भी कोई स्टूडेंट दिल से रिस्पेक्ट करता है, सहज भाव से आपके मन से, क्यों डॉक्टर साहब, ये नहीं निकलता गॉड ब्लेस यू, ये इसकी बहुत वैल्यू आपके सहज भाव से निकलता है, आप कहें ना कहें, आपके हृदय से निकलता है, आपकी आत्मा से निकलता है.
इनहेरेंट फीलिंग सर, इनहेरेंट फीलिंग
होती है. वो तभी होती है अगर स्टूडेंट हम्बल है, स्टूडेंट एथिकल है, स्टूडेंट कनेक्टेड है ना. सो इसका रिजर्वायर इकट्ठा कीजिए, बहुत जरूरी है इसका रिजर्वायर. बिसाइड्स द नॉलेज, नो हाउ, नेटवर्क, रिस्पेक्टेबिलिटी का और आशीष का, आशीर्वाद का, पंजाबी में कहते हैं असीस दा, reservoir इकट्ठा कीजिए, which you can get it in cash anytime, every time, anywhere, everywhere. So that is very, very important, which is somehow I see dwindling. Student कुछ बन जाता हो, कहता कि it was because of me the teacher is not, कई बार once in blue moon it happens like that. So never ever let that attitude remain with you. Belong to your country, जैसे मैडम ने कहा था, बिल्कुल सही है. Belong to your institute, belong to your mentor. आप कह दीजिए ना, जो कुछ भी हूँ मैं अपने देश की वजह से हूँ, अपने इंस्टीट्यूट की वजह से हूँ, अपने मेंटोर की वजह से हूँ. मेंटोर कभी नहीं रखेगा आपका क्रेडिट अपने पास, वो हमेशा अपने स्टूडेंट को अपने से ऊपर जाना देखना खुश होता है. जैसे टीचर, जैसे पिता देखना पसंद करता है अपने बच्चे को अपने से ऊपर, उसी तरीके अपना एक टीचर पसंद करता है. बट द स्टूडेंट शुड अप्रोच द टीचर लाइकवाइज. अगर करता है तो बिसाइड्स द एकेडमिक नॉलेज, नो हाउ, इंडस्ट्रियल नो हाउ, ये सारी चीजें के बाद QBD, PAT, सब कुछ, इसके अलावा जो ब्लेसिंग्स आपको मिलेगी, जो डिवाइन बेटिट्यूड मिलेगा आपको, उसका कोई साने नहीं. There is no parallel to that, and that will be the cementing force with your knowledge and know-how, and which will culminate into enormous kind of accomplishments. That's what I personally feel. Thank you very much. Thank you. Anything else, Dr. Dr. Sharik?
I think from the chats that we've been seeing in terms of faculty response, I think they are appear to be blessed by all the wisdom that all of you have really provided them in a nutshell. And it's a very informal conversation. Sometimes you don't
I
hear
see some emotional kind of
in typical structured presentation, you don't get that opportunity also for speakers to kind of have that conversation. But I really like to thank Dr. Bhoop for kind of setting this up as an expert panel session. It has served two purposes. One of them was a more of a technical kind of a summaries thing, and also as a guiding session, because a lot of the people, I think all of them, they see yourself, Dr. Patel, Dr. Vandana as their heroes, Dr. Shirsagars in their life as a role models for some.
One comment is, "Sare Bhagwan mil gaye mujhe is session mein."
Absolutely. I think that
आपके अंदर ही है, अपने आप के अंदर देखो,
हमारे अंदर ही
खोजना पड़ेगा, वैसी वाली बात है,
खोजना पड़ेगा.
हाँ, अपने अंदर ढूंढ के पा जा जिंदगी वाली जो शेर है, वही पूरा हो गया.
लेकिन हाँ, अगर गाइडेंस मिलती है तो तभी
बिल्कुल सही बात. I think you really have, all of you have really shared your perspective on the valuable inputs in life. Or as a researcher, as an academician, life is not very easy, as Madam said. You go through ups and downs. But अगर आदमी के अंदर conviction हो, और अगर you have that grit and perseverance, definitely quality research one or the other day definitely comes back and it pays you in terms of recognition, in terms of projects, in terms of appreciation. वो है, लेकिन उसके लिए वो एक सफर हमें तय करना पड़ता है, and for that you have to be ready, I think. So I think in summary, I would say that, crystallizing what all of you have said. So I think for some reason I don't know, I'm very worried Dr. Ijaz has not yet joined in. He had confirmed to me last night when I spoke by email. But I hope
he's not responding to your call as well.
He's not responding to. He doesn't have a WhatsApp on his number. I'm just trying to call him other way.
You don't have his number?
I have his number, but it's not. There's no WhatsApp to it. So I'll
WhatsApp नहीं है उनका. उन्होंने he exited out of WhatsApp, because of the privacy, so-called privacy issues.
Okay. So I'll have to dial him from another platform, and I'll see. Hopefully, I'm concerned because he's very particular in terms of his replying back. He immediately replies back. I told him it's like 5 o'clock, sir. It's 2:30 and 5 a.m. for you in Eastern East Coast times. I said, is it okay for you, or should I delay it? He said, no, I wake up at 4 o'clock in the night anyway. So. I'm hoping that all is well at its end.
By that time, Dr. Vijay Sagar and Dr. Muralidhara,
yes,
probably, they were not there. They can add some
wisdom. So I think the Dr. Sagar, there are a lot of students or folks from industry as well. Your piece of advice to them, number one, and number two, sir, one thing that I've seen. When I was in US and when I moved back to industry, I think Dr. Vandana Patrawale and LD Patel are kind of an exception, but there is a lot of synergy between industry and academia. Faculty is going on sabbatical, the industries regularly come for some work. So I was at University of Maryland. The University of Maryland has very close to US FDA. All the IVC work was pretty much done at our lab 10, 15 years back. So a lot of synergy there. They come in, a lot of interactions, faculty go to industry. That somewhere is lacking. I think this industry is doesn't trust academia. Maybe they think they won't be able to deliver. And academia feel that industry has just not treated as the right way. So वो sync नहीं होने की वजह से, I think there's a lot of talent at both ends which is not getting synced. So Dr. Sagar, on your decades of experiences in industry and also interaction with academia, where do you think we're lacking?
Yeah, doctor, you asked a very pertinent question. And when I look back, say last 10, 20 years, the interaction between the industry and the academia was at a minimal extent. But today I am saying the interaction of the industry people with the academia has increased substantially as compared to what we were there. But I don't see it other way. I don't see that academicians coming to the industry and getting themselves exposed to what happens in the industry.
Right.
I strongly feel that for every pharmacy teacher, and I'm sure the industry will mobilize, if they get exposed to in the industry for about say 3 to 6 months and try to have the practical aspects of how the industry works, they will be very influential and very effective teachers. So I'm coming back. I propose that for every pharmacy teacher, there should be a necessity of go to the industry for about 3 to 6 months.
Right.
It can be called as apprentice or it can be called as anything. And I'm sure the industry will support it. I don't see any reasons why they will not support it. Even some industry will volunteer to compensate also suitable. But I think now industry is trying to come close to the academia. Like I never interacted with the industry, but today, fortunately, I got an opportunity to interact with professors in the BCP. There is one local college at Vashi also where I'm interacting with them. And they are inviting. So, but I don't think it stops there. It has to be other way around also, that the academy also needs to come to the industry. So that's what I want to add here. Thank you so much.
Doctor, sir, you are unmute, sir. Sir, unmute yourselves.
Dr. Muralidhara, can you further add some words of advice, wisdom for the young participants? Maybe due to QBD management, software usage, in general.
So, okay, so now that you people have spoken, there's nothing to add from my side. The only thing I keep saying myself and my kids and my so-called, I do teach here and there in management schools, be it IIT, some IIMs, but I say that one thing is statistical thinking. So coming from a background of an engineering stand, and I'm not a hardcore statistician. I keep saying it every time so that I can learn more and more. So, but statistics, the way it's going at is the kind of essential tool or essential need, just like we learned basic maths, algebra, geometry, and keep that bend of mind. Statistical thinking is very essential at whatever stage you are in your research or academics or else in your teaching perspective, and leverage technology. And I know that some professors want to go with the conventional way of teaching. I think it's a time for us to get exposed to a little bit of flip model of teaching. And with the pandemic, it's going to help you out. And from a management perspective, I think we are all paid for problem solving. And how do you use statistics to solve your problems? That's where you start thinking from the first principle. Whether you're in pharmaceutics or quality control. So I'm look at the best part. I'm a textile engineer. I'm learning a lot of things from you people. Every time I interact, I keep talking like chemistry. I talk to the chemistry, IT department, or the pharmacy department. I'm talking to industrial engineers, management students. And this is the best part of my profession is because I keep my antennas open. That's what my professor used to say, keep your antennas open. And try to learn from others. I think this is what we talk about Medici effect in Harvard also. How elephants can learn from mosquitoes. And that's the Medici effect. Probably I would have used the different words there. But how can I learn from a person who is non-statistician or working in a pharmacy department from best practices? So जो सीखना बड़ा चोर होना चाहिए. Start stealing others people. You don't want to reinvent the wheel. So just steal knowledge from people like Bhupender. He is a Professor Patraule. Just their tanks. Open it. Kitna bharo. Tumhara jitna bhi hai plate bharo. Khalo. So that's what I keep saying myself. Just be open to yourself and be curious. That's a very important word. Curiosity builds capability. This is, I put it on my top of my desk. Curiosity can only build capability. Yeah. Thank you so much, Professor, for the opportunity.
Maybe from academician's perspective, can I add something?
Definitely, madam, please.
Yeah, so in fact, I had made this presentation for Vaibhav Summit, when Prime Minister invited speakers from all over the world to interact with Indian scientists. And the major thing which industry wants is data integrity, where, we academicians, I would say many of us do lack. And as I mentioned earlier, how many of us actually, bring out the chromatograms and look at it before the students tabulate the data. Second thing is industry always works in a time-bound manner. So they want the deliverables to be given on time, which if university shakes hands with the industry, it becomes their duty to do it. So in the beginning itself, they say, if you want, you keep more time, keep a gap, but later on, don't give us reasons and excuses. Third thing is they want us to have a market perspective. Why are we doing whatever we are doing? That should be absolutely very, very clear in our mind, which at times doesn't happen. Also, for translational research, I would say in India, there are certain points to ponder, like, are we modifying rules or rewriting rules every time we wish to collaborate? So do we have written norms? So many of the colleges are lacking there itself. We do not have ready drafts of confidentiality agreements, MOUs, which can be shared with industry. And then when industry comes, if it is multinational, we share something else. If it is local domestic industry, we share something else. Do we have norms again laid down in terms of intellectual property? What kind of royalty should you agree upon? Whether it would be 3% of the gross or of the profit, and whether it will be one-time payment, milestone payment. We do not have any written norms for any of the milestone payments. Also, when we are working actually in research, we are not maintaining many times log books for the instruments, equipments. So all these are, sort of provisions of data integrity. We are talking about CFR compliant instruments. How many institutes can boast that they have CFR compliant and they can maintain the whatever data is there will not be tampered? That is another thing. We do not have GMP setups. Now we have taken a very big step ahead. ICT is coming up with GMP setups. So in case we want to go for clinical trials, we are again dependent on third parties, and your research actually gets nipped in the bud if it doesn't go to the second, third, higher level. So other major important thing is, can we ensure confidentiality from student side? You have signed CDAs, but are students who are going to graduate from your institutes, they'll keep it confidential? Have you signed any CDA in turn with them? That is what we do in our lab. So that, they know that they are legally bound and they cannot open their mouth to other industry about this particular problem. Again, we are failing to take some clinicians' perspective. And that is one reason why our lab is moving little fast forward, because any problem which I initiate, I first take their perspective. Not only that, actually we have to take adequate survey from end user also. For example, I was working on a rectal microbicide preparation, and we do not have sufficient devices there. So those who are using it, we have to take them into confidence and take surveys from them. And most of it, why we as academicians fail is because of our very huge ego. So we need to dissolve our ego. I think these are the points which were suggested. I suppose I thought that let me share them.
Fantastic, sir, madam. You really
Good,
nailed
very
it.
good.
You nailed it, as they say. Following up on your comment that you mentioned, I think one thing that you really emphasize is having now a very multidisciplinary approach or a translational approach. So sometimes I see academics that they're very siloed into their work. They're very fixated about their own domain. They hesitate going beyond their domain, see an application. So I think going forward, I think that approach is pretty much dead, as you mentioned. And so we last 2 months back, we did a FDP, a very similar of this on artificial intelligence in healthcare. So if you look at all these Center for Excellence in Artificial Healthcare, half of them are physicians, half of them are statisticians, half of them are engineers. So a crosstalk between them into what is a problem statement from a clinician point of view? How do that translate as an engineer into a code that helps me? And how does that translate back to a solution to a clinician that can understand that language to him? So all that, I think, has to be built in. I think that only then, I think, Dr. Padrale can kind of comment on that, and she does a lot of translation work. So I think your thoughts on that.
Very rightly said that it is the multidisciplinary approach which will take us, to the next level now. Now that AI and machine learning is really coming in, as Professor Boop knows, in the morning session also the speaker emphasized on it. And in fact, I asked a question, from student side, as to if AI and machine learning is coming, just like some of our participant has asked, what is it? Which jobs will, pharmacists not able to take? And which are the jobs where they should focus? But Professor Bhoop came to my rescue and he twisted the question to get the right kind of answer. And that was, what we should actually have in our syllabus. What more can we have so that our students become ready for tomorrow? So yes, indeed, we need to look at these, because very fast we are moving towards all such kind of systems where AI becomes a part and parcel.
Part of also life, absolutely, madam. I think with the FDP that we had, I think it's penetrating pretty much every aspects of a life. Healthcare is no far behind. Assisting physicians, prescription medications and everything. So because of the huge computational power that is coming in and the technology that is building in, there is going to be a transformation, I think. And at Dr. Bhoop's message is, how can I bring value to the table, and how, in terms of my creativity, in terms of my vision. I think that's where the machines cannot copy you. That is your uniqueness, and that is where we will be defined going forward. So if we are just doing Me Too work, if we are just doing regular stuff, obviously there is a lot of there is a problem with us then. But if we define ourselves in terms of uniqueness, then definitely there are opportunities, and rather more opportunities, because the machine then augment you rather than compete with you, I think.
Yeah, very rightly said that the machine can augment you, machine can extend you.
Right.
Machine has artificial intelligence, and we have natural intelligence. So that artificial intelligence cannot be surrogate. Can be surrogate, but cannot be superior to that of the natural intelligence. So it has to be complemented. Many things we cannot. Our neurons are limited, and we have kept them limited. Very novel laureates have 13 to 14% of their neurons active. Right. And a saint has much more than that. And it's usually said that. Absolutely. But normally 7 to 9% of the neurons are really, really dynamic. But so we have to complement with the more viable, more efficient machines like which are based upon AI. But AI is based upon natural intelligence itself. So we must understand there is no surrogate to human beings. We have to master the machine. Absolutely. Not machine has to master us. In software too, very, very often, my students also, tend to quip, sir, मैंने कुछ नहीं किया, sir, ये design expert ने किया, ये JMP ने किया, sir, मैंने तो कुछ नहीं किया. और मैंने कहा, Gigo is valid. अगर garbage out आया है, तो garbage must have been in. गया. So it is so समझना बहुत जरूरी है. Right. So is Dr. Ijaz in?
No, sir. I'm very concerned for him. He hasn't.
I think अगर अब नहीं तो फिर
pause कर लेते हैं. हाँ, so we'll stop our program here, and then we'll continue with the valedictory at 4:15 or so. I'm very concerned, hoping everything is well with him. We can definitely reschedule his session. I think a lot of people are waiting for his session, so I'm just concerned. Whether madam, you were saying something? I think
yeah. In fact, just taking the discussion forward, just as Professor Patel has indicated, we have tendency. I'm saying we because we teachers have tendency actually to not encourage our undergraduate students to ask questions, and that same thing is percolating down. So I think if we give them some simple problems in the class also, which they can tackle, they can come up with innovative solution. For example, when I take cosmetic lectures, you believe me, I take the simple, whatever is there in the syllabus, then I cover the advances which I have come through, and then yet open up the forum to them that come out with a particular cosmetic formulation. And out of 30, we have only strength of 30. Out of 30, at least 1 or 2 will come up with a patentable solution. That means that is the power of those young minds. So we need to just open up their parachutes, which I feel we are not doing. So
we're not doing
a lot of responsibility relies on us as teachers.
Yes, and I think you nailed it, the point that the ego comes a lot of times in between. Sometimes we are not ready for the questions that student asks us. So with the fear of not being able to answer them, we stop them even from asking questions. I remember, when I had joined University of Maryland, it's a very interesting cultural shock for me. At day one, when I was there, my cousin was in his last year of PhD, and he was doing with a very renowned scientist in protein chemistry. So he took me to his lab, and we were sitting in his lab, and suddenly his advisor, Ron Giles, I still remember him. He walked in his lab, and this guy was Berkeley graduate, shortlisted for Nobel Prize. And both of them are having conversation as if they are two friends. And my cousin is telling, Ron, I don't think that would work. He's saying, no, Nazim, let's look at the perspective. And that was my first day in US, and it was a cultural shock for me. So the level of conversation, I think we need to have a lot of openness. I understand we have a certain level of hierarchy in our culture as such in Eastern countries, but that should not prevent us from letting the students open up towards us in terms of their curiosity, I think. Dr. Bhub, I think you are muted, I guess. Sir, आप म्यूट पे हैं, आपकी आवाज नहीं आ रही.
आपने बिल्कुल सत्य कहा है, very rightly told, we need to be, one-to-one communicator with the students. I keep sharing with the students, I cannot take class where there is monologue.
Absolutely, sir.
It has to be, if not multi-log, at least dialogue. एक बोले है ना, dialogue. It should be, not unidirectional. It should be bi-directional. So if they are not asking questions, मैं उनको कहता हूँ, अगर आप नहीं पूछेंगे तो फिर मैं पूछूंगा.
मैं पूछूंगा.
So आपको पूछना है, So prompt करते हैं, पर लेकिन many of them, tend to be in their, ivory towers, or tend to be hibernating in their mind. So, you cannot prompt everybody, but that should be the way. I keep telling my students 5 P's. 5 P's. मेरे जो research students होते हैं. First P, perspiration. उनको वो लिख लेते हैं कई बार ना. और when I became the chairperson of the department, उन्होंने मुझे ना frame करके एक पेंटिंग दी जहाँ पे वो 5 P's ही लिखे हुए थे, मुझे भी दे दिया, है ना. Perspiration पहले, मेहनत. Number 2, passion. You have to have passion, enthusiasm. Number 3, perseverance. You will not be getting everything what you aspire or aspire for. So you'll have to overcome obstacles. Number 4, patience. The results will not be obtained in a day. Patience चाहिए. And number 5, not the least, not in that order. 5 is piousness, piety, integrity. जिसको Dr. Vandana कह रहे थे, honesty. Unless you have all these 5, you cannot be, a full-fledged researcher or scientist. फिर भी आप कभी ना कभी में आपको दिक्कत आएगी, या पेशेंस में, या परसिवेरेंस में, या परस्पिरेशन में, या पैशन में. Then there is panacea, that is prayerfulness, prayer. And then last P is, 6th P. P is prayerful. Sit in one corner and harvest that cosmic energy and ask for, oh God, oh Allah Ta'ala, oh, Bhagwan, mere oh Vaheguru, मुझसे पैशन मेरा जा रहा है, मैं ये प्रॉब्लम को ओवरकम नहीं कर पा रहा, मैं पेशेंस नहीं रख पा रहा. So, it is granted. If we teachers, will not interact with the students, who else will? And मैंने देखा है कि parents की बात students शायद ना माने, teachers की बात का जरूर महत्व रखते हैं, मान रखते हैं, है ना, ज़्यादा रखते हैं, है ना. So उनको रोल मॉडल मानते हैं. Parents को कोई विरला को रेयर मानता है रोल मॉडल, बट टीचर्स को फिर मान लेते हैं, उनकी बात मान लेते हैं. सो हमें उनको ये बताना कि प्रेयर इज पनेशिया, सो ये सारी 5 P's के अलावा तो everything is granted. Prayer is, harvesting your own inner energy into reality. So कोई ऊपर वाला बैठा है कि नहीं बैठा, हमारा अंदर तो बैठा है ना, उसका रेप्लिका तो बैठा है हमारे अंदर, हमारा उसको ही हार्वेस्ट करना, sit in one corner and get try to connect with the same, and the results are remarkable. That's what everybody of us must have experienced. So that needs to be shared with the younger generation, I personally feel. So काफी बातें off the track हम कर रहे हैं, है ना.
बहुत अच्छी बात हुई. I just got a call from Dr. Hussain. He just there were some technical issues at his end, so he might take some time. So we have rescheduled his talk after our voluntary program. So so if you permit, sir, then we can stop the session out here with a thanks to the entire panel.
Let me share that Dr. Ijaz Hussain is the probably most meaningful speaker amongst we all, because he's a blend of drug regulatory affairs, the academia and industry experience. He brings brings with him all the pertinent experience. 1 decade, 1 decade, 1 decade, 10 साल teaching, 10 साल drug regulatory, and 10 साल 10-12 साल उन्होंने industry में भी बताए. And he was at the helm of the affairs when FDA instituted QBD guidelines. So please, all the participants must not miss his talk.
Yes,
after
yes.
the
So
webinar.
4:45. So 4:10, 4:15 we'll be starting the voluntary, and by 4:45 he's agreed to join in. He was really apologetic for the inconvenience that has been caused, but definitely for the person of his stature, we accommodate that. It is a genuine concern. So, sir, with your permission, then we'll close the session here. We'll keep the meeting on, and by 4:15 we'll start a formal voluntary program. And by 4:45, we will have Dr. Jas Sain coming here. So for all the participants, just as a reminder, as Dr. Bhup mentioned, is a very valuable resource person, and we need to really think for him as a whole bird's eye perspective on the entire quality initiative. So thank you once again, sir, on behalf of all of us. I think it was a fantastic session that really put वो कहते हैं पूरा आइसक्रीम की टॉपिंग हो गई है, टॉपिंग ऑन द आइसक्रीम कह लें आप उसको केक के ऊपर. With this informal chat, sometimes these pearls of wisdom for us, sometimes these are one liners, for some it becomes a change in their life. It completely changes the entire direction of their life. हम शायद चले जाएंगे, लोग हमें याद करें कि वो सर ने ये बात की थी, पटेल साहब ने ये बात की थी, वंदना मैडम ने ये बात की थी, शिवसागर सर ने ये बात की थी, आज मैं यहाँ हूँ करके. I think it happens a lot with our teachers, and we have students coming after years together and say, sir आपने वो बात कही थी. So this might be an half an hour, an hour investment from our time, but sometimes it changes the entire course of direction for a lot of people. I'm sure that, and I pray for that, that we'll have more Dr. Bhoops and Dr. Patels and Dr. Shirsagar and Dr. Patravales coming in to take this baton from this generation to the next generation to a next level of excellence. And as our Honorable Prime Minister always says, we need to create that kind of sustainability within our own research system, where we don't have reliance over others, but rather we are self-sustainable, I think. So that is the goal, I think, to which we should strive forward. Thank you once again, sir, for all of you. It's really a pleasure and honor for you to hosting. Thank you so much once again. So for the participants, so we will keep the meeting on here. And by 4:15, we will start our voluntary program. Dr. Vaidya, who's the executive chairperson from ISTE, will be joining us as a chief guest. And we've also invited Dr. Bhupender Singh, who will be also joining us. We'll have a short voluntary program, and post-voluntary program, we'll have a talk by Dr. Ijaz. Thank you once again. Thank you so much. Thank you, Madam. Dr.
Thank
Patel.
you, ma'am. Thank you, Vanda, ma'am. Thank you, sir. Bhup, sir. Thank you much.
Thank you. Thank you. Thank you, all participants. Please stay tuned. You can either be there in the meeting or you can join back at 4:15 for voluntary program. Thank you.
Thank you, sir.
Thank you.
Good evening, Ejaz sir.
Good evening.
Good evening, sir. Sir, what we have done now, we have scheduled your session post validatory program because that program was already being scheduled, no?
Yeah.
So it will start at 4:45 something. We'll start with your session and we'll be validatory program. Is that okay?
Yeah. What's the time now?
It's 4:15, sir.
It's 4:15. Yes.
Good morning. Good morning. Assalamu alaikum to Dr. Jazz.
That's good.
Thank you, sir, for joining in. No issues. What we've done is we've rescheduled things. So if you want to log out back and then join at 4:45, that will be fine also.
Yes. All right. Sounds good.
Thank you. Thank you so much. Take care. Exactly. Good afternoon to all our participants. So we're just waiting for one of our guest speaker to join in just within a minute or so, and we'll get started. Thank you so much for your patient waiting. Thank you.
Sharik sir.
Yes, sir.
Our director sir has joined. I think we should start the session.
Sure. Okay. Okay. Please go ahead. Thank you.
I welcome all the participants to valedictory session. So for valedictory, we are fortunate to have with us the Dr. Bhupender Singh Bhup, Professor, University Institute of Pharmaceutical Science, Punjab University, Professor Vijay D. Vaidya, Executive Secretary of ISTE, Dr. Abdul Razak Hanudgi, Director of Anjuman-e-Islam Kalsekar Technical Campus, Dr. Sharik Sayed, Dean School of Pharmacy, faculty members, and all the participants from different prestigious colleges across the India. I extend my warm welcome to all of you for the valedictory of this 1-week AICTE ISTE refresher program. Before I invite the guests to address the gathering, I request our Dean School of Pharmacy to give the overview of all 6 days session. Dr. Sharik, it is over to you.
Okay, thank you, Professor Abu Sufyan. So, Honorable Executive Secretary for ISTE, Dr. Vijay Vaidya Saab, Dr. Bhupendra Singh Group, our invited guest for today, our campus director, Dr. Abdul Razak Hanudgi, and all the dear participants. I think you will all agree with me that it was a very exciting and a very enriching week for all of us. The entire week that was dedicated on quality by design. I think, and I'm very confident that it really served the justice and the purpose of it. And the outcome that was defined was the participants should get a good awareness about the concepts and principles of QBD in terms of its implementation, in terms of its challenges. So I think we were very fortunate to have a whole plethora of speakers with us who did an excellent job in kind of giving us a very good overview of quality by design in terms of its concepts, in terms of implementation, and in different domains, be it analytical sciences, be it drug delivery sciences. We had an exciting session just an hour before. We had a very informal panel discussion. We had all of the leaders in this field. We were fortunate enough to hear from them in terms of their perspective, the challenges of QBD. So all in all, I think I'm very excited and very happy that something that we had planned for, and we did reach that certain level of outcome that we intended for. So I think all of the participants, based on the feedback, also would have agreed that it was a very worthwhile use of the entire week in terms of our learnings of QBD. So I'm personally very, very thankful to all the speakers who have given their very valuable time. Please know that all of them are extremely busy people who have given us valuable time in helping us understand this extremely important concept of quality by design, which to all of you would agree has had a major impact on Indian pharmaceutical industries. And because of that, the credibility of Indian pharma industry as a global drug provider has really gone up, and we have made that mark across the world. So I think it was a very valuable FDP. And I'm very positive that all of the participants, there are a lot of research scholars in India, there are a lot of faculty participation, would take this forward and push this science beyond what is being done till day to day. So without taking much of our time, I would like to invite our first invited guest, Dr. Bhupendra Singh Group. He doesn't need any introduction. He's been a mentor and an advisor to our faculty development program. I cannot, I don't have words to express my gratitude, heartfelt gratitude towards this noble gentleman who's been considered as the guru of the QBD world. 300 plus publications from his group, #1 group across the world, considerable clout across the industry where everyone goes to him for advices, but a very humble, down-to-earth person. You call him at 11 o'clock at the night, he'll pick up your phone and he'll answer to it. So without further ado, I would like to invite our first invited guest for the day today, Dr. Bhupendra Singh Group, who's an architect of this faculty development program, single-handedly helping us to get this thing set it up. A lot of the experts that I've talked to have, whenever I've told them that we are doing this FDP on QBD and Dr. Bhupendra Singh's group has recommended your name, the first line that came from them was, Bhupendra Singh Saab ne keh diya, to hame aana hai. So that kind of networking and connections that he has across academy industry, I'm personally very, very impressed by his credentials, both as an academician and as a humble person. So without wasting much of our time, I would like to invite Dr. Bhoop for his thoughts on this FDP. Sir, over to you, sir.
Thank you so much, Dr. Sharik Said and Dr. Sheikh Abu Sufyan and Dr. Abdul Razak and others. I'm so privileged to be amidst this prestigious platform. In fact, this is my third appearance. First I delivered the talk, the inaugural talk. Then, we were there for this long plenary session, and now thank you so much for making me part of this valedictory session as well. In fact, that was I keep a very busy profile thanks to my students, thanks to my institute, and thanks to the aspirants, who tend to learn from whatever we have learned in the last several decades. But due to your enthusiasm and zeal and passion, which I saw in Dr. Sharik, and um, and the Kotri. So I succumbed to his pressure, and I became part of this very useful program. And when I saw that zeal and late night exchanges of the conversation till 11:30 or sometimes 12 a.m. we had been communicating about the potentialities of various speakers, and thanks to those speakers who kept that honor of being trusted upon, and they became part and parcel of this program. Very useful, certainly very useful. I saw the feedback. I attended a couple of sessions as well, besides of course my own. So very useful sessions those were, and very eminent and meaningful personalities, because I see so many, because QBD being talk of the town, I see some semi-quacks also appearing here and there who tend to claim themselves as QBD expert, but we do not have any hesitation in listening to them, but as long as they are providing the factual information. But that is not the case, because they are rather misleading the scientists. So all the scientists roped in over here by Dr. Sharik and his team, have been real experts in their respective domains, and that's what makes the real difference, because it's not institutes and the conferences are not built by buildings or by the big sponsorship. They are made by the faculty, the resource faculty, who tend to deliver the goods. And I if I name one and leave others, I'll not be doing justice. Everybody, be it the senior ones like Dr. Vandana and Dr. Patel and Dr. Vijay Sagar, or be it the juniors, everybody did commendably fine. And for that, I give credit to not only the speakers, but to the platform which was provided to us by your team, Dr. Sherry, you and your, it's very well knit team and very efficient, starting from the brochure, which was very professionally designed, and to, using this platform where there were hardly any lapses, of a Zoom platform, and, fantastic. I must say, kudos to you, and very useful. But the purpose is that the knowledge and the know-how, delivered over here, that should be carried forward. That's what, as a humble teacher, I aspire for. That let the trainers be trained. If one teacher out of this is, duly trained, he can train 30, 40, 50 further, more students, So if we are able to together, able to train and stimulate even 20 minds rationally, I think that's much more than enough we have achieved. But I'm sure not only 20, I think hundreds of minds have been stimulated. How fervently, that time will tell. The aspirants will tell in due course of time, if they tend to implement, if they tend to apply, if they tend to replicate, if they tend to continue learning and propagating and applying the same. Thank you very much once again for providing me this opportunity. And I wish all the participants and the organizers many more such, ventures of learning and applying. Thank you so much.
Thank you very much so much, sir. It has been really an honor and pleasure to be working with you. You are truly an inspiration to all our faculty members. So thank you once again for staying, sir. Professor Abu Sufyan, over to you.
Okay, thank you, sir. Going ahead, we have the chief guest of the today's program, Professor Vijay D. Vaidya. It's a great honor for all of us to have Dr. Vaidya with us, who is Executive Secretary of the ISTE. I take this opportunity to introduce Professor Vaidya. Professor Vijay D. Vaidya has obtained his B.Tech degree from Karnataka Regional Engineering College in 1981. He worked in the field with MSFCon Mumbai at different sites in the Karnataka, Andhra Pradesh, Goa, MP, etc. He joined teacher in the field 1985 as a lecturer in the civil engineering department in government polytechnic. Since then, he worked in a different capacities, including the lecturer in civil engineering, head of civil engineering, training and placement officer, vice principal, and principal. He has worked in administration in Directorate of Technical Education, Mumbai as assistant director, worked as a deputy secretary in Maharashtra State Board of Technical Education, Mumbai. He is having the long list of achievement in his credit. Few of these are the Government Polytechnics Kolhapur got accredited by the NBA during his tenure of the principal. He is the pioneer in designing G-scheme curriculum for the MSBT. He developed online monitoring system of the institutes for MSBT and is still in practice for deciding the performance. He has written the book on valuation, attended various national and international conferences. Professor Vijay D. Vaidya is the Executive Council member of the ISTE since 2012 and he is presently serving as Executive Secretary of ISTE. He activated various programs and activities as per the policy of Honorable President and Director of the Executive Council. Professor Vaidya is working hard for development and progress of Indian Society of Technical Education. On behalf of Anjuman Islam Kalsika Technical Campus, I extend my warm welcome to Professor Vaidya and take this opportunity to invite him to give valedictory address of the program. It is over to you, sir. Sir, you need to unmute yourself.
Thank you, Sheikh Abu Sufyan.
Welcome, sir.
For my detailed introduction. Really, it was a great introduction. Good evening, everybody. It's my pleasure to be with you for this valedictory program of multidisciplinary and translational drug development organized by Anjuman Islam Kalsika Technical Campus, New Panvel. It's always my pleasure to be with you because whenever I meet you or whenever I actually visit your campus, I learn a lot of new things, particularly Mr. Burhan Harris, Executive Chairman of Anjuman Islam. Really, he's a nice personality. Dr. Abdul Razak Honnudgi, Campus Director, and the development of campus has been done under his able and dynamic leadership. Dr. Sharid Sayed, Dean and Coordinator of this program. Really, I would like to appreciate the efforts you are taking for continuous 3 programs. The resource persons you are gathering from, who are having national as well as international recognition, and that is a really great thing you are doing. Then Dr. Bhupinder Kumar Bhu, special guest of today's function, and members of organizing committee, and my dear participants. Basically, this is a AICTE ISTE sponsored program, and here it is expected, or the objective of this program is to give knowledge about the emerging technologies trends to faculty members or to enhance their knowledge, so that will be helpful for them in their regular day-to-day working or while teaching or dealing with the students. And I think the way in which all 3 programs are conducted, the resource persons and all, and the topics selected, I feel that the objectives of our programs are fulfilled to the full extent. You can say that 100% objectives of our programs are really achieved, and I would like to congratulate this Institute and their team for this great success. Now, if you consider the present scenario, you are all are aware that the technology is changing at a rapid rate. Each and every area or each and every part of our life, there is change in technology. And another important aspect is that whatever technology we were using yesterday is becoming obsolete today. And whatever technology we are using today as a latest technology, we know that it is going to become obsolete tomorrow. So such a rapid change in technology is taking place, and under this situation, it is very difficult to predict what is going to happen or what technology will be there after 15 years, 20 years, or something like that. And if you consider in that direction, we can see that there are many emerging areas, and they will govern the total technology field. They are artificial intelligence and machine learning, IoT, cyber security, then blockchain technology, 3D printing, etc. But it's very difficult to predict exactly how the things will be and what will be. And under such circumstances, it is very difficult to predict the types of jobs that are going to come. And when we are not in position to uh, we are not in position to identify the types of jobs that are going to come or the technology that will be used, it's very difficult for us to train the students in such an area. So for this, the basic requirement is that our teachers should be well versed with the new trends and technologies that are coming and that is being uh, changing continuously. And for this, they have to take continuous training or they have to update their knowledge. One thing you have to make sure that if you don't update your knowledge, the rate of change of technology is so fast that in due course you will become obsolete. And once you become obsolete, you know what will be your value. And unfortunately, even in your institute also, you will lose your value. So in order to maintain your level or you maintain your value, it's highly essential that every teacher should undergo continuous training and he should ensure that he is moving at par with the technology. Now, when we say about all these things, the question a teacher may ask, yes, sir, I am ready to go undergo different types of training, but who will give me an opportunity to undergo the training? Where can I get the training? So here AICT and ISTE comes into picture. You will find that AICT is having tremendous schemes for the betterment of teachers and students. So there, if you will see that there are different schemes, just like different types of training that is also there for teachers. And in addition to that, there are different schemes for individual teachers, and individual teacher can get benefited by those schemes. So I feel that if you want to get all every information, we have a habit of visiting website of AICTE, and you will come to know about the new initiatives at AICTE, and definitely that will help you to develop yourself. Even at ISD level also, we are doing a lot of things for the betterment of teachers and students. We are organizing different types of programs, and continuously we are having different activities. And one latest activity which we are going to start in short period is that we are going to arrange internship training for students. Internship training for students. It is an online training program where in which the students will be given training about the internship, and we also will assist them for getting good jobs, and at the same time this will be a totally free program. So that program we are working on that, and we are likely to come with that program shortly. So when such different opportunities are there for the teacher, it's our duty to enhance our knowledge and be with the time. Another thing is that nowadays, everybody is talking about the national education policy, and I feel that as a teacher, we must be aware about the national education policy. Now, to am I audible?
Yes, sir, you are audible.
Just there was some problem with the audio. So in national education policy, whatever drawbacks we are observing in present education system, they are mostly covered. Because when we talk about the present education system, we talk about the different drawbacks. And the main drawback we talk about is the outdated curriculum, or the curriculum is not at par with the latest technology. But here in national education policy, it is clearly mentioned that curriculum should be curriculum should be revised, it should be relevant, interesting, and updated regularly to align latest knowledge requirements and to meet specified learning outcomes. So that is expected that curriculum should be modified continuously and with latest knowledge. So this is our duty to ensure that the curriculum is implemented at a regular interval and frequently with new trends and technology. Most of the important thing is that the objective of national education policy is to see that India will come out as a global educational hub. That is the basic objective. Means when you say that it's a global education hub, it is expected that students or faculties from outside India, they should join our institutes for different types of programs. Means presently we see that our students are going abroad for higher education. And in national education policy, it is expected that this part should be reversed. Means instead of our student going outside students should come to our institute. And when you expect such thing, the basic requirement for this is that our institute should be of a global standard. So our institute should be of global standard and our teachers should be of a global level. When such thing happens, that is global level institute and teachers are globally competent teachers and institute, definitely the product will be globally competent. So that is the basic requirement, and I think it is the duty of all we teachers to ensure that if we want to be in the race, we have to modify ourselves, we have to modify our institute, we have to develop our institute so that we will move with time and technology expected. Another thing is that in national education policy, the emphasis is given on the research. So I feel that it is our duty to encourage students for different types of research and to develop and to get the different types of patents, and that will definitely help to enhance our institute. In addition to that, now this is an online platform, and I think this is the platform due to which I can see that there are 151 persons have joined this program today. So that is a really great thing. And another thing is that whatever resource persons they were invited, they were of a eminent nature and they were from the reputed organizations or institutes. So really this is only possible with the help of this online platform. Because if this program would have been arranged in a face-to-face mode, I don't think maximum you can allow 40 to 50 participants, and at the same time such kind of resource persons would not have joined this program due to their inability to spare much time. So here, this is the advantage of this online platform, and the same advantage is identified by national education policy. There, they are insisting on the blended type of learning. Means now we are thinking of face-to-face mode. Only due to this COVID pandemic, we have shifted to online mode, and even the COVID pandemic, we have successfully completed our teaching and examination last year, that is 2021, and we have seen that no student has lost his year. So here, the same thing, this online platform is of a tremendous use, and national education policy says that you should use the blended learning method, and it consists of blended learning is nothing but you can have some classes in a face-to-face mode and some portion in an online mode. And this actually this implementation has already started, and recently UGC has come out with the notification that they are proposing 60% face-to-face mode and 40% in online mode for their programs. And for that they have asked for the suggestions and from the faculties and different suggestions and proposals for that. So I feel that this trend is going to come and we must be ready to face such challenges. One thing you make it sure that during online platform, it's very difficult, means you cannot make your lecture interesting unless and until you use ICT tools. Because ICT tools are the major things with which you can make your lecture interesting, attractive, and you can convey or communicate the message you want to convey properly. So I feel that teachers should be well versed with the ICT tools. So these are the new trends or new things that for which we must be ready. And I feel that teachers are also aware that they have to face the future challenges and we have to develop each and every one. And another thing is that if we develop ourselves together, definitely we can achieve a lot of things. Particularly this institute, Dr. has done a lot of things in past also, and they are doing a lot of initiatives for the betterment of teachers, students, and particularly Dr. Abdul Razak Pandutgi is a dynamic person, and uh, whenever I meet him or whenever I have a discussion with him, I really feel that he is always thinking for the betterment of his campus and his students. So really, such a dedicated person can do something for the betterment of teachers and students, and definitely, and he is having strong support of his management for all these activities. And it's always a pleasure for me to visit their institute, and particularly I still remember their hospitality. Due to this online program, participants have missed that great opportunity uh, to see their hospitality. But I think in future you can visit their campus on behalf of them only. I am asking you. You can visit their campus, and they are really good people, and they will give you good hospitality. And then once again, I would like to uh, congratulate Dr. Sharik Sayed for his tremendous, dedicated and sincere effort. So really, you have done a great job. That the programs in which you have the programs you three programs you organized, particularly getting eminent persons and that too from abroad. That is a great thing that shows your dedication. So I really appreciate your effort, and I feel that this program, all these three programs are conducted in a best possible manner by your enthusiastic team and dynamic leadership. So I feel that you will be with us for different various activities for the betterment of technician education, and together we'll move ahead. With this, I feel I have whatever I want to express that already have expressed, and I wish success for your future activities. Thank you.
Thank you, Dr. Vaidya, for your unconditional support. I think your support has been really a bedrock of whatever that we're trying to do here. Without your support, none of these three programs would have been possible, sir. Your constant support and your motivation is the driver for us to come up to the expectation that you have set for us. Thank you so much, sir. Over to you, Professor Subyan.
Thank you, Professor Vijay D. Vaidya, for your inspirational valedictory address and valuable guidance to us as a teacher. Going forward, we have with us the Honorable Executive Chairman of Board of Institute of Navi Mumbai, Mr. Burhan Harris. I take this opportunity to invite him to address the gathering with few words. Burhan Harris, sir, it is over to you. Sir, you have to unmute yourself.
Good afternoon. Assalamu alaikum. Namaste. A bright day today. These days, yes, it has been so bright and so hot. Warm is not the word. But it feels. It's so bright. It feels so good. I thank every host, every guest. Sorry, to begin with, every guest to be inviting, to be sort of attending the sessions with this whole complete webinar. It is undoubtedly a grand job done by my team. Zach is here, Sharik is here, and of course behind the camera people. Wow. I mean, it was all done by heart, from heart. It was all done with extreme intensity and feeling of doing it right and doing all that is required to be done. We are all in the business of education, spreading it, giving it, spreading it, and giving it in the best manner, not best possible, but best manner. Giving it the way it should be given. I repeat this. It's a noble job, of course, everybody says it's a noble job. And it has got nothing to do with commercialization. This is my belief. This is my team's belief. This is my institution's belief. This is my Anjum Islam's belief. From top to bottom, to an office boy to the president of Anjum Islam, this is what we feel we have to do. All that is we are doing. Because it's more, because it is giving knowledge, spreading knowledge. It has to be the attitude. This has to be the attitude to be doing this job. Well, we all know that it had been 1.5 years. We're going through tough time. I mean, doing anything, everything in these times and for the last 1.5 years is so tough, so difficult, almost impossible. And yet, we are doing, even in these times of 1.5 years, every activity that we did was appreciated, highly appreciated by every guest who attended. We have so much more to do. We have so much more to do. But as Dr. Vaidya said, that only because of COVID. And this is serious. This hurts now. I mean, it was a joke sometime, but only because of COVID. But now it has become, it is something which is hurting. We have all got so much to do, and we're ready with it. Spreading knowledge, spreading with perfection, spreading with quality, spreading the way it has to be done. And that is what we want to do. And thank you every guest, and my hosts, the team of hosts, everybody who was involved in managing this, is arranging this and managing this show. Thank you so much. I salute everybody for understanding the importance of attending and understanding the importance of doing your job in these kind of jobs of spreading knowledge and spreading it right, spreading it in the best possible manner. And that's what we have been doing. And yes, thank God that it has been appreciated by him. It has been always appreciated. Thank you, Dr. Vaidya. At least I heard your words for the organization and for its team, for the institution, I mean, for the campus and its team. And see, it is also the appreciation, which is part of important factor to spread. I mean, whatever work we do, ultimately, if it is appreciated, it adds the quality of performances by everyone who is involved, and it does wonders with the job. And that's what we've been doing. It's a service. It's a service by the team. It's a service by the people who came, who spoke, and who even appreciated. Thank you so much. There's so much more, but I have a bad habit of talking so much. So I've learned. I'm learning these days to stop myself, to put a stop to myself. That's it. Thank you.
Okay, sir. Thank you, Burhan Haris sir, for your motivational words as always. Your guidance and motivation keep us going. Going ahead, we have with us the founding director of the campus, Dr. Abdul Razzaq Hanudgi. Sir, you have inaugurated this refresher program. So on this occasion as well, I request you to give concluding remark of this validatory program. Sir, it is over to you.
Thank you, Professor Abu Sufyan. Respected guest for today's valedictory program, Dr. Vijay Vaidya, Dr. Bhupinder Singh, which I still remember during his inaugural address, that keynote address, it was mesmerizing, and as Dr. Sharik said, rightly said, many participants, they took part in this program because of names like Dr. Bhupinder Singh and all those resource persons, be it Dr. Ijaz Hussain and the spectrum of speakers drawn from various parts of the country and under different expertise. As I said, Dr. Ijaz Hussain, Dr. Ranjit Barsikar, Dr. Vandana Patrawale, Dr. Vijay Kshirsagar. So these are the people who are, who already established their benchmark in the field, and identifying, connecting with such great resource person, connecting with our participants is really a great job done by that organizing team of this faculty development program. Headed by Dr. Sharik Sayed, Professor Abu Sufyan as a coordinator, and all their faculty members. As rightly said by Dr. Vijay Vaidya, without the support of a management represented by a person like Burhan Haris, we just can't do any such activities. So I know, despite his health problem, I think nobody has observed, nobody has realized that he spoke so well, and he was not well, but still he made it a point to attend this valedictory program, because he knew the importance of this program, and only to encourage the team, and also to tell us go ahead with all good things. He always makes it possible to attend the program. And I think anything what we say, either in terms of thanking or paying our gratitude to Dr. Vaidya, so our words always goes, very less, and we cannot truly express our gratitude just by words to Dr. Vaidya, because he is a person who is always associated since inception of our, this campus. And that way, persons like Dr. Vaidya, our president has declared that the persons who contributed, who always remained an inspiring pillar for developing our campus, they're always our brand ambassadors. So Dr. Zahir Kazi, who was there during inaugural program, so he always used to say that the persons like Dr. Vaidya are our brand ambassadors, not only for this campus, but for entire Anjuman-e-Islam. Because we know the amount of support, the amount of encouragement, and even whenever we were going wrong, and immediately he used to remind us, Zach sir, just say this is how you need to be corrected. See, such a frank person who does such a good handholding, and this particular faculty development program, which is funded and sponsored by AICTE and ISTE, was purely due to the initiative taken by Dr. Vaidya, and he asked us, out of a couple of proposals sent from our campus, he asked us which proposal to be sanctioned. Say from IST headquarters, the capacity of being executive secretary, he asked us which one you want. So this is the way he has got connect with our campus. Sir, you took that initiative, and this pandemic has become a blessing in disguise, that 1 week on-campus program got split into 3 online programs, and according to your expectations, all these 3 online programs were conducted so systematically and with complete dedication, and invariably every program was attended by not less than 150 participants. And that too participants, they are not, very interestingly, if you can I was going through the report, there were in the first 2 days only, there were 450 plus registration for this QBD program. And that too participants not only coming from academic institutions. They were not only faculty members, even industry persons and many reputed scholars from premier institutions like NIPER, IIT, Drug Controller Office, Pfizer, Sun Pharma, and so many. From such organization, the response came to this FDP, and out of 450, we could restrict only as per the requirements, a particular set of number. So this shows how important such faculty development programs are, and the way it is organized, planned, and the topics and the source persons, as you said, really hats off to my team. And I must mention a point to all the participants, particularly for Dr. Vaidya, Dr. Ejaz Hussain, and also our chairman. Our chairman knows that. See, during this program only, our dean, Dr. Sharik Sayed, he met with, his entire family almost escaped from death. His entire building caught fire in the midnight at 1 o'clock a couple of days back. And it was a hell of a job. In that entire building caught fire, and they escaped almost by the grace of Almighty. They escaped from that kind of situation, and immediately they got shifted to his cousin's home. And even then, his spirit of being associated with this FDP, so that did not go down. He did not put any excuse that I have met with my family, met with this kind of situation, and he did not just hand over that responsibility to someone and just got away. So this is the level of commitment. Hats off to Dr. Sharik Sayed. This is the reason why, always God comes to our rescue. And situation which I studied about that fire, it was really a miraculous escape. And God is great and kind enough to all of you. And this is what the commitment level of a campus, sir. So
thank you.
I must thank Dr. Sharik and even coordinator Sabu Sufyan. I did not make him realize that okay, this FDP organization gets affected. No, he took the lead, and the commitment of Professor Abu Sufyan. Again, hats off to you. And these are the people, and the team of School of Pharmacy is setting itself as a benchmark for our entire campus. So in future, we have got huge plans to make that our campus will become really a lighthouse for industry. Those kind of ambitious plans we have got, and in such endeavors, people like Dr. Vijay Vaidya and all resource person with whom we associated, will definitely add value to our efforts, and we will be always in contact with you. And you must be knowing that being executive secretary of ISTE, Dr. Vaidya, his commitment level, no doubt, nobody can, match what he did. So for the last 1 decade, he is associated with ISTE. He was an executive council member and subsequently became executive secretary of ISTE. And the moment he took over as executive secretary, so immediately, see, his best quality is immediately putting his responsibility into action plan, and he adopted various measures, because the people, unless go through the details, micro details, so one cannot do justice to the expenses part. It's not just giving money and telling institutes or any organization to conduct the program. So even he resorted in a good amount of measures to reduce expenses. And parallelly, he activated a large number of programs and activities as per the policy devised by Honorary President of ISTE, Dr. Professor Pratap Singh. He's one of the very eminent personality in our country, Mr. Pratap Singh Desai. So he has got full support and confidence in Dr. Vaidya, and that is the reason why ISTE could take a lot of programs and activities, and also very democratically under the directives of the Executive Council. So people like Dr. Vaidya will definitely make or bring meaning to the organization wherever they are associated. And when such a person associated with our organization, then we are very fortunate. Sir, thank you very much for being with us. And I know all participants, the amount of interest, though it's an online program, but that thirst for learning will make all of you to gain the things, from those resource person. You attended this week-long program, and I'm very sure you did not attend only for a piece of certificate, but you attended for gaining, enhancing your knowledge. And that knowledge, when you become trainer, when you start spreading this knowledge, say amongst your students and other colleagues, then only the effort put by this program will gain a meaning. So I wish all participants to take further whatever you have learned and try to devise some ways and means so that you propagate all that you have learned. So that is what my appeal on behalf of the entire organizing committee and our management of Anjuman Islam. So thanks to all participants being with us until this time with all that commitment. So finally, I just, out of our curiosity, I just put one link here in the chat box. I request all of you to express your experiences or whatever you felt about this FDP in one single word. We would like to collate a word cloud based on your responses. So one link I'll be putting in chat box. So kindly click on that link and try to express your feedback with a single word. So let me put that word. The link I'm adding into the chat box, and I will be sharing. Professor Abu Sufyan, can I get?
And in the meantime, can I?
Yes, sir. Yes, sir. Yeah, please, sir.
And individually share a thank you to the guests and the speakers of today. Yeah.
Yes, sir. Yes, sir.
Dr. Ejaz, thank you so much. And we will be leading you time and again. It's not the end today.
Actually, Dr. Ejaz sir will be speaking post this valedictory program because of some technical issue. His program got rescheduled after this valedictory.
Sorry again.
So there are a lot of people who are waiting to hear him. He's a guru in the quality by design. So
he can hear me. Yeah.
Yes, he can hear you.
Okay. And the other guests?
Yeah, they are done. Everything is done. That's the only
program. Thank you. Thank you so much. And booking you for the future without the date, of course.
For sure. For sure.
for all the speakers, wherever we feel that you are the right person, we will be shamelessly inviting you. Thank you so much.
Thank you, sir. I think definitely participants are waiting for Dr. Ejaz Hussain's, a final session, which is very important. And but still, if I see the words that are coming from all participants, now that speaks, yes, we did justice for the program. And this is what Dr. Shariq, Professor Abu Sufyan, and all faculty members, organizing team. So all that credit goes to you. Everybody is telling excellent, outstanding, fantastic, insightful, fabulous, good experience. It is a wonderful program. And you can see these are the words that inspire all of us to do, again, good things and take good initiatives in the future. So thank you all the participants for having such a faith in us and providing your one word, feedback. And this will definitely help us in devising our program. Okay. So with this, I will close my concluding, remarks. Once again, thanking all the persons. Thank you, Dr. Vaidya. Thank you, Dr. Ijaz Hussain and all the participants. Thank you, Burhan Saab.
Thank you. Thank you, sir.
Let me close this session. Okay.
Thank you. Thank you, sir, so much. On behalf of the entire team out here, I would really like to thank Dr. Vijay Vaidya for honoring this occasion for us, Dr. Bhupender Singh Bhup for the entire mentoring and FDP that this has been constructed just purely because of his guidance, sir. I really thank you for all your support.
Thank you,
Dr.
I
Vijay
saw
Vaidya,
you the first day also, Doctor.
Yes, a fantastic person, fantastic academician. And also like to thank our management of Anjuman Islam, Mr. Doran Harris, who's representing that management, who's always been a supportive for any initiative. Our very own campus director, Dr. Abdurazak Panutagi, has been a very proponent of quality initiatives like this, and he's been very careful about this, a very strong proponent of it. So it's an honor and pleasure to be working under his leadership here. And I also like to thank my entire team out here and the lead coordinator, Professor Abdul Sufyan, and along with him, the entire faculty coordinators, Professor Maria Lal, Professor Salman Beg, Professor Madhuri Gaikwad. All of them have worked tirelessly to get this program up and running with no glitches as such, and the entire 6 days were flawless as such. Going forward,
Razak Fayazuddin is saying something in the chat. Okay, sorry.
Okay, so I think without further ado, I think we'll move on to our final last exciting keynote address, for which it is an honor and pleasure for me to be inviting and introducing Dr. Ijaz Hussain. So Dr. Ijaz Hussain holds a bachelor's and PhD degrees from universities of Mumbai and Cincinnati, respectively. Has 35 years plus of unique experience, and that covers a whole range of domain that includes academia, US FDA, industry. And US FDA he's acted as Deputy Director of Office of Pharmaceutical Sciences, US FDA, Vice President and Global Head Pharmaceutical Development at Sandoz, Chief Scientific Officer at Philip Morris International, Chief Scientific Officer at President Biotechnology at Wockhardt, and President of National Institute of Pharmaceutical Technology Education. In his advisory practice, he focuses on developing corporations, individual professionals as self-authored architects. A very interesting line that you spent, of the evidence that they need to make sound decisions efficiently. He engages with science companies on challenging problems in product development, manufacturing, quality control, and assurance. He's also He's a member of several boards and also serves as an adjunct professor for Long Island University. It is truly an honor and privilege for me to be welcoming Dr. Ijaz Hussain. I will remember this as one of the key moments of my life, to be introducing such an accomplished scientist, a person of such an authority, to our forum. He's been very humble enough to accept our invitation. A lot of people are waiting to hear from you, sir. As people have told Dr. Bhupender Singh also, that you've been a chief architect of bringing the whole concept of quality by design philosophy from more of places like Stanford research to an implementation in form of US FDA guidance and percolating across industry. And I might say, and you might correct me for that the place which Indian industry, pharma industry occupies is primarily because of the quality framework that you and others have built into it, so that our products are recognized worldwide, our generics are recognized worldwide as quality products. And as they say, 1 out of every 3 pill that is consumed across world is produced by India. That's a proud moment of us, and we couldn't have done. The Indian pharma owes a lot to you, because if the quality is not inbuilt, especially in medicine, there is no way out of it. So I think, sir, you've been a towering figure in this whole world of quality by design, and making sure that gets implemented across the board. You've been a champion of that, sir. I mean, it's so long that it'll take probably another half an hour for me to just introduce, but I'll give it and have you speak, sir. A lot of participants are waiting to hear from you.
So much in one man, my God.
So much in one man. It's just an honor and privilege for us at Comsec Technical Campus to be welcoming you here at this virtual forum. Over to you,
meeting you, wonderful seeing you. Thank you.
Thank you. Thank you. Namaste and salam to everyone. There was no technical glitch. The glitch was I overslept. My usual time of waking up is 3 o'clock, and I did that, 3 a.m. And then waiting for 5 a.m. to talk to you, the waiting put me to sleep. I'm so sorry about that. My apology.
That's okay. That's fantastic.
So I will be brief, and I know I'm following Dr. Vaidya and others. I really appreciated listening to those discussions. So what I would like to do is think about the teacher, and I think if I could share my screen, I'll start with that. So I'll take you back to several years. We all are good regulators of pharmaceuticals, not just FDA, and I think that's the key message. And I wrote this article in 2016 on LinkedIn, April 5, 2016, after visiting the Government College of Pharmacy in Aurangabad. And I've been, from 2014, I've been speaking at American universities, at Minnesota, Iowa, and others, on good regulators of pharmaceuticals, but I give the credit to Professor Bhoop, whom I think who invited me in 2014 to Punjab University, educating the next generation of pharmacists for industry. And thank you, Professor Bhoop, because you set me on a path to really thinking about the professional development. I think I also, around that time, the University of Cincinnati gave me outstanding alumni award. And at that moment, I had to remember my teachers, from Professor Bhalla, Professor Tipnis, Professor Manik, to my own PhD advisor, Professor Richel, at that time. And I think the key is the dream begins with a teacher who believes in you, who tugs and pushes and leads you to the next plateau, sometimes poking you with a sharp stick called truth. And I think that was the motivation of that. But at the same time, what I would like to do is this. I think one thing which I learned there, I am from Kendriya Vidyalaya system, central school system in India. And I think the key is Vidya and Avidya, I think the central school motto was, "Oh knowledge, do remove these covers so that we may see thee," I think was the key. Now, quality policy, the school maxims on the wall, but not in our heart. And how do we bring that in our heart and not? So I just wanted to sort of give you that sense. This is where I started thinking, and I'll switch to a few slides that I want to share with you, and not take too much of your time. And that is, I had, as soon as I was invited to speak, I put my slides together and shared it immediately. So I can assure you, my technical glitch was not my preparation. It was all prepared, but I overslept. I apologize for that. So I thought what best to do is to how do I let me just, between hello and goodbye, between stimulus and response, I think is our space to respond. And in that space is our freedom and our opportunity. And I think that's what I wanted to do, is to share with you, because as I'm a bachelor of pharmacy from India, and formulation design, formulation development was at the core. Pharmaceutics was at the core of my passion. And so when I got to the US and FDA, I was told that all this pharmaceutics, industrial pharmacy, pharmaceutics is a black art. We don't care about it. And I'm not joking. So because we have a regulatory system which approves pharmaceutical products, but doesn't want to acknowledge pharmaceutical product. It goes with approving a new drug, a generic drug. And that historical ignorance of pharmaceutical science is the problem that we face today continuously. So I think when I had an opportunity to challenge the prevailing regulatory system, that just knowing a drug is not enough, you need to understand formulation and you need to understand quality by design, was my opportunity to help see clearly now. So I created a Vision 2020 on November 28, 2001, of what is quality, quality and performance by design, continuous real-time monitoring of quality, specification based on mechanistic understanding, how formulation and process factors impact product performance, high efficiency and capacity utilization, real-time review and inspection from Rockville, White Oak, New Jersey District Office, and so forth. Now, where in COVID, we have no choice but to have real-time review and inspection. So I think my journey, which I chronicled on LinkedIn, Schrodinger's cat, and my journey from 2015 to 2020 was my own professional development. So to see clearly now, and I think where I want to take you is building on Dr. Vidya's thing. We need to self-empower our own professional development. And just to remind you, 2001, what was the year 2001? And if you look at the dates I'm talking about, October, November, what was happening? The world changed with 9/11. And with the last name Hussain and the birthday of September 11, I'm leading the FDA efforts on this. So you can imagine what was on my head. And I was on every no-fly list at the same time. So I think that was a tough time emotionally and in every way. So 9/11 changed it, changed the world, and now COVID has changed it again. So one aspect which was very important for me, which I learned being part of the system in US, was a symposium that was held in October 2001 by the US Command and Control Research Program of the Office of Assistant Secretary of Defense. So that was the first time I realized that how we make sense making, meaning making is a process. And we don't learn how to do that in a structured way. We learn it ad hoc. And so mind matters was the key. Your beliefs are cause maps that you impose on the world, after which you see what you have already imposed. Common sense is a collection of prejudices acquired by age 18. Common sense is not so common. So how do I bring the logic and scientific rigor to my sense-making process was the key. So if you think about it this way, let's start in the physical domain. We interact with objects and events in the real world. And that means we experience. Experience is to feel emotions. Experience is practical contact with events and objects in the world. So we have an object. So when we interact with an object or event, we have a data representations. We go away from the physical domain to information domain. And the data gets to informs us. And we have a situation awareness, which is individual, and then it has to be shared with others. So we move from information domain to cognitive domain in our mind, and where our awareness interacts with our mental models, our prior knowledge, and we get in that interaction, we gain understanding. From that understanding, we make sense. And from making sense making is a process. And when we make good sense, we arrive at good judgment. And then we have our intention, or we have an intent command, which goes to planning, directive, synchronization, action, and the cycle continues. So in this cycle, emotions, beliefs, and perceptions are essential. This is what gives us a passion. This is what gives us the motivation to do things. But it is a double-edged sword. Emotions, on the other hand, and beliefs and perceptions take away from truth. Because we are biased, we have conflicts of interest and so forth. So how we learn to manage our experience to make good sense is the key. And my practice in the industry since then has been developing decision tools to for improving situation awareness and effectively utilizing meetings, interactions, day-to-day interactions in a corporation or to respect, to appreciate emotions, which is experience, to make good sense. So meeting management, so it's as simple as that, to build an evidence architecture, how you manage your communication, how you articulate, how you write, how you speak, how you meet, how you take meeting minutes. This is as simple as that. So that's what I focus on, because we have the mindset, quality, cost, and time. You can only choose two. No, you can choose all three. You can have quality, reduce cost, and reduce time. And that was the mindset I just wanted to share with you. So the key to that is integrated design and systems thinking and feeling. So how do we integrate feeling into design and systems thinking is the key, because all we do when we do that is we go from pity to sympathy to empathy to compassion. So without feeling, a scientist who can't feel is the most dangerous person on earth. I'm absolutely clear. I think the debacle we do is the scientists follow the science without feeling is disaster for the world. And I think we are experiencing part of that right as we speak at the moment. And the key is by design is by intention, according to a plan, life spiral, by heart, learning to listen, design thinking is that fundamental. And we don't do that. Follow the science without that feeling is utter nonsense. I think, and what we are seeing is the debacles all over the world of how we have responded to many aspects of the crisis that we are in. And I think the key there is intention is the way I see it. I think you start with intention, niyat. I mean, you learn from your grandmother. My grandmother says, niyat saaf rakho. If you have your intentions clean, that's the starting point to quality by design. And I think so as this is a memorable picture for me. It was ICH negotiation 2004 in Kyoto, Japan, where I was signing off on ICH Q10, and I was acknowledged for 3 words: design space, Procrustean, epistemological. Procrustean is one size fits all. If you don't fit, I'll chop your legs. And we are Procrustean most of the time, and that's the lowest order of consciousness. And we have lost epistemic trust in a large percentage of the population. The yearning in the collective consciousness of human majority for equivalence and sameness in how powers of a society regard and treat us is a major driving force in how future manifests. In the context of quality, equivalence is of the same consciousness. This is what I wrote in 2015 on LinkedIn. And so if you're following me, you have seen that. Inside that, again, during a recent visit to Japan, was the reason. And the key lesson there was when I was doing a training on culture of quality in Ratlam in Madhya Pradesh, I was asked to give the lecture in Hindi. And I said, I can't speak Hindi. I have to speak Hindustani. But speaking a different language allows you to make better meaning of the language you speak in English, for example. And when I did my training, culture of quality training in Ratlam, the major plant for IPCA, and doing it in Hindi was that door opener. I said, I'm making better meaning now. And so I think as Indians, leveraging our languages, leveraging our diversity to make better meaning of what is a big opportunity. And I think India has that advantage, and it has to take that advantage there. Seeing is believing, but knowing is in observing. So why is FDA processes of observations, Form 483, so important? Because seeing is believing, and believing is not enough. Because believing is biased, believing is unjustified most of the time. Knowing is observing, and observation is what we observe is what we think we want to observe. So cognition and emotion and intention, I usually change the word intention, because if we are intention, our cognition and emotions interact and we see what we want to see. So skewed perceptions have to be addressed there. And I just want to remind you, we are makers of meaning. And if we can learn the process of making meaning, sense making, I think, and acknowledge that in our professional activities and respect emotions, I think we can do better. So quality by design is nothing but connecting the dots. I think if you look at compartmental modeling in pharmacokinetics and apply that to your evidence from drug substance. So each section of CTD-P2, for example, critical drug substance variables and relate to formulation development. So how does section P 2.1.1 in drug substance relate to section 2.1.1 to 2.2.1? Essentially, that's what the design thinking is. This is the evidence architecture. So how do you connect connect the dots is the key. We have to be better than placebo in the real world, but our better than placebo evidence comes from clinical trials when we have to double blind everybody. We don't trust anybody. So pharmacodynamics of placebo is something very, very important. And by the way, the evidence is very clear. The price-response relationship is the same as the dose-response relationship. I'm not joking. These are the hard evidence that we have. So one interesting editorial is Pharmacodynamics of Placebo, which was published in Neurology in 2015. So we have an uneven playing field for generics. In US, brand has direct-to-consumer advertising, but in the real world, generic means cheap, means low quality, warning letters, import alerts, US population context. What it means is generics are laden with negative emotions and negative expectations, and therefore automatic substitution to generics starting in 2014, we could measure it that 60% of heart patients, when the color and shape of a tablet changed, they stopped taking the medicine. So the whole notion of a cost-effective generic got lost because we didn't manage expectations. And the key is quality is what? Quality is subjective, is conscious experience, is qualia. Experience means to feel emotions when in contact with things, and learning from experience is the way forward for all of us, especially the teachers, because in 2010, the Institute of Medicine came out with a paper saying that the healthcare education system in the US has utterly failed. I'm not joking, those are their words. And because the third leading cause of death in America is the healthcare system. And I'm not joking about that. That's a study from Johns Hopkins, which is a local university here. And if third leading cause of death in America is the healthcare system, this is before COVID. God help us what happens next. And so in 2018, the Institute of Medicine, the Institute of the Academy of Science and Academy of Engineering came out with a workshop paper which says the way forward is integrated design and systems thinking and professional development, which is experiential, not sitting in a class and studying. That's not continuing education. Professional development is experiential learning, and how we learn from experience is the key. And I think we have a tool inside under good practice. How do you feel inside? Most of the time, as growing up as children and as teenagers, we want to look good, and then we want to be good, not rock the boat. And at some point, we get internally validated, self-validated. We have the assurance, we are self-authored, and then we want to do good. And then some point, we mature, we want to see good. Between stimulus and response, there is a space. In that space is our power to choose our response. In our response lies our growth and our freedom. What response you choose is your suitability and capability. We have at minimum two good. So everything we have in pharmaceuticals, say for example, 21 CFR to 11.25, education, training, and experience is at the minimum necessary to work in the pharmaceutical factory. But we have never defined what is education, training, and experience appropriate for each job, for each level of complexity. And we want to make sure to give assurance to others, one must first be self-assured. And fake it till you make it is not an option in the pharmaceutical sector. So suitability is maturity, capability is development, and you have to be self-authored, and you have to stop saying, "Tell me FDA what to do," and wait for FDA inspection and warning letters to do something. You have to be self-authored. You have to be problem-solving, system orientation. Did I achieve my goals? Follow a process methodology. Fourth order of consciousness, which is a term which I have used, is to be self-authored in every job, every line worker to CEO, and be problem-solving. So that order of consciousness concept comes from Professor Keegan at Harvard on constructive development theory of adult human development. And I've seen this across. So for example, I have done studies of Pfizer and Amgen here. So Pfizer in November 2001 came to FDA and say, we have adopted a don't use, don't tell policy for PAT, process analytical technology. And in 2004, Amgen started, if I don't look, there is no problem. And that means they were not doing effective root cause investigations. And What was holding them back? The change prevention system and feeling system. We are immune to change, immunity to change, and we have to solve problems and we have to prevent errors. So the orders of consciousness in a corporation and in human beings goes from third order being socialized, fourth order being self-authored, and fifth order being prevention of errors, problem solving to prevention of errors. So we have to manage three systems: knowing system, change prevention system, and feeling system. And change prevention system, the regulatory system itself is the change prevention system. But at the same time, the mindset we have is, it is FDA approved, it is validated. So if I don't look, there's no problem. That is the level of order of consciousness we have all around. And I think we have to increase that order of consciousness. That's really the message. So between stimulus and response is your choice. Choose wisely. Some adults continue to develop, but at different rates. Many do not. Teachers, professors have no other option to keep developing themselves. And to keep developing themselves, they have to learn from their experience, not just formal education and training. Learning from experience is a wonderful opportunity, and that's where my focus is. So I just want to stop there. I did not want to go into details of how did the PAT-QBD journey begin. There is a video that's available. You can see it when I recorded at FDA. But the point I want to make here is this. QBD and PAT was my own development. The process understanding, suitability and capability of a manufacturing process. I learned to be a human, and it was my journey to know what it means to be a human so far with this. So I stop with that, and hopefully this message is useful for you to take home. And I really apologize for the so-called technical glitch, which was me. Thank you for listening.
Wonderful. Wonderful topic. I mean, congratulations for the selection of the topic. And
thank you.
And wow.
In fact, each time listening to Dr. Ajaz is, like sitting as a student in the class of a learned teacher, Dr. Ajaz is an inimitable blend of experience in industry, in drug regulatory affairs, and in academia. More than 1 decade experience in each discipline. So he's, quite unique in his experience, and whatever he shared through his experiential wisdom each time, he shares newer experience. His presentations are available on various sites, particularly on his own website, on his LinkedIn platform. And each time, unlike me, his presentation is, filled up with newer and newer ideas. And it was he who was at the helm of the affairs. He shared that during that time, when the suffix Hussain was very difficult, to sustain in US, he made, landmark, achievements, as far as the QBD implementation was concerned, because he was at the helm of the affairs in FDA at that very time. So listening to him is really a delight, and it's a matter of privilege for not only me, for everybody over here, particularly the young aspirants. It's a when I connected with Dr. Ajaz, it was for me a dream coming true kind of experience. And since then, we have been interacting with each other, and each time it is I who's the beneficiary. And I think the young participants over here would also be benefiting a lot. Thank you so much, Dr. Ajaz, once again for reconnecting. Thank you so much.
Thank you
for being so humble to be available for such relatively modest platforms. Right?
No, not at all. Dr. Vaidya, thank you. I enjoyed your talk very much.
Really, it was nice interaction with you, sir.
Same here.
Yeah, it was really interesting.
Sorry, I was disconnected in between, so I could not connect you, sir, at the end of your session. So thank you, Professor Dr. Ejaz, for giving insightful session. Your session was very wonderful, and we have got good insight of the quality of design, sir. Thank you.
Thank you. Thank you.
Thank you, sir.
Thank you. Assalamu alaikum.
Sir, I could not see any question in a chat box because I was disconnected. So earlier chat got erased. So if someone else can just read out the chat on my behalf. I hope there is no question.
Yeah, no, I don't think I don't see any questions. Sorry.
Only praise it.
One question is, can you correlate QBD in coming years as the game changer?
Yes, I think, see, so quality by design is by intention. So by design is by intention, and quality is qualia, which is consciousness. And I think we are at the cusp of two directions. One is good and one is evil. I mean, good and evil exist in our society, and which direction we choose, good and evil, will be determined by quality by design of our thinking. And I'll be very, very frank and open. I'm concerned that we may choose a pathway for technology and diminishing the value of humans. And one of the worry I have is the way FDA and others have jumped on Industry 4.0 and PAT-based continuous manufacturing is for one purpose: eliminate humans from manufacturing. So eliminating humans from manufacturing is just a easy way out for assurance. And the best way out for assurance to get assurance of quality is to develop ourselves. I think we are at the cusp of diverging in two paths, good and evil, as simple as that. And if we don't pay attention to quality by design, we will get up into an evil end. That's the way I see it.
There is one.
According to you, Dr. to make the things better understood by the younger participants. So, according to you, means quality by design principles can be taken up in either way, in good as well as evil way.
Yeah, it's intention. So by intention, right?
Yeah, quality by intent.
So quality by design is a very effective and efficient way of getting to your intention.
Yeah.
The question is your intention good or evil?
Further, this is an era. Even in India, we are seeing big companies switching over to continuous manufacturing. So in this case, majorly it is artificial intelligence, virtual reality, programming logic circuits, which are outweighing, and the human component is hardly there. Once, yeah. So what role QBD can play in such continuous manufacturing?
Very, very important question. I wrote a paper for PHARMA Times in India on that digitization, and about a month ago or maybe very recently, because you may remember my academic research was all artificial neural networks. So my beginning as academic in this come from artificial intelligence background.
Wow.
Quality by design started because I was working on computer-aided formulation design while in Cincinnati, correct? So it was an artificial neural network. I was a bit ahead of my time then because I came to FDA to build artificial neural networks. And by the way, they have developed a few networks to detect cheating by design at the moment. So that's happening. But where I come from is with the smartphones to smart factories, we have dumbed down humans. Humans are dumbing themselves down with our smart technology. Mm-hmm. The smart technology is not making us smart. And that is the danger that I see. So what I did was I converted based on my natural language processing and machine learning to point out that we can turn around, don't let social media addict and dumb you, convert Facebook to the book. When you're on Facebook, remember the book, not the face. And so what I have done is with my LinkedIn and Facebook, I have used my social media as making contemporaneous notes of my experience. And that is my learning. I don't let social media make me dumber. I use social media to get smarter. That's the key.
It's not the technology which should act as the master of the human being. It should be the otherwise. Human being should master the technology. That is the message to carry home.
Yes. So by the way, IO is I was born in Hyderabad, so from South. So IO is a word which I remember. IO, the smartphones are getting smarter than me is what you remember.
We are so happy. We are so happy about it, you know.
If you permit, Dr. Bhoop, can I ask a question? Since you're moderating the way. So, Dr. Hussain, I mean, with this whole advent of computation technologies coming in, a lot of computational power coming in, you know, artificial intelligence becoming more and more smarter at using, more and more human as such, you know, how do you see the future of the, you know, scientists and people who are budding researcher in terms of their niche areas where they should invest their time, energy, and expertise into it, so that tomorrow they don't get obsolete, but rather become expert, develop those expertise in this area. So thoughts on that, since you've seen a lot of these technologies from very close.
So very good question. Very good question. So the reason I'm pushing for self-authorship, because so with Professor Mala Menon and some of my classmates from Bombay College of Pharmacy, we ran a program called Academia to Industry, disconnect to filling the gap between academia to industry. So we are running some courses there, and so what we are emphasizing is self-authorship. If you are going to follow a standard operating procedure, why can't you write that procedure? Why can't you take responsibility for writing it? I'm not joking. I get paid almost $1 million to write a response to a warning letter from FDA. I said, what nonsense is this? You can't write a response to a warning letter? Self-authorship starts. So I've been pushing Bombay College of Pharmacy to think about how do you do labs is to self-author the procedure that you would, or improve the procedure when you do a lab. Self-authorship is the key. That means, so remember, I think so, therefore I am. Descartes, the Enlightenment, they fooled us. I feel, therefore I am. I am, therefore I am thing. So put feeling first, because intention comes first, right? So the Enlightenment and the Enlightenment mechanistic thinking has fooled us. I think, therefore I am is incorrect. I feel, therefore I am. I am, therefore I think. That's what we have to do. So you cannot compete with artificial intelligence. So following an SOP is, forget, if you're training to follow an SOP, you will not have a job starting 5 to 10 years. You have to be writers of those jobs. You have to be creative. You have to take ownership. You have to write the programs, what needs to be run, not be programmed by that. That's what I would suggest.
Yes, I think that really nails it down. Our own campus director has a question: how does the confluence of big data, artificial intelligence, and high-speed internet has on, in general, the impact of that? How is that confluence affecting the overall life and pharma industry as such?
So I have been at the top of the pharma companies. Okay.
Right.
And I've been at the top of FDA in quality. I feel a danger. The orders of consciousness at those levels are self-authored, but so egoistical. Profit is the name of the game. And the difference, so the where we are is corporations have merged with the government. Instead of being public servants, I have been very critical of FDA. The way they have made decisions in this COVID are shameful. I'm talking about US FDA. And the acting commissioner is my direct boss, Janet Woodcock. So I have the duty to tell her, if the orders of consciousness of these individuals doesn't go to a higher order, we are doomed. We are enslaved by the big data and so forth. So instead of a utopia of leveraging big data and so forth for humanity, for developing humans, we will enslave humanity. And remember, economic GDP and Human Development Index, United Nations, economic growth happens because of human development, not at the expense of human development. We are at that junction right now, and whether we respect human development and bring economic growth for human development, or we focus on economic development at the expense of human development, we are at that junction right now, and the next 10 years will define that. And unless we step up to emphasize quality by design for the right reasons, intentions, we are in trouble.
Absolutely, absolutely. So with this whole, as you mentioned, this whole issue, speaking to FD about your concerns, and I think that the whole entire pandemic, during this whole year of pandemic that we had, I think pharma industry as such has taken a big hit in terms of ethics as such, in terms of profitability, in terms of providing value-added, affordable medications to more of an exploiter, you might want to say. The whole classic case of remdesivir in India, at one point of time.
What an amazing shit! That was such a.
I've known people who have sold everything, sir. Their whole, the wife jewelry and stuff for the 10,000 rupees vials, 5 vials, 50,000 plus black market went to 1 lakh. It was pure water. It was just been rolled out. ICMR overlooked it, unfortunately. So we're getting a bad rapport here.
I'm fully aware of it. I'm fully aware of it. I've been monitoring the situation from each and every corner I could. My family members have survived, at least that's the grace of God, that they've survived the ICU experience in India. But what I have to tell you is this, I lost my mother. My mother and I were among the first to get the COVID. I survived, my mother did not. And my wife, who's a physician with Johns Hopkins, she wrote a paper, nobody should die alone. And I think if I can do anything for the rest of my life is to honor my mother's promise that I won't let people die alone. And if anything that we can think about is letting people die alone, that is the uttermost shameful aspect that we will remember for the rest of our lives after this.
Yes, sir. Yes. I think as a pharma industry and as people who are working into pharmaceutical sciences, I think we really need to take a very hard look at it, essentially, with regards to the whole question about ethics and in terms of what we are doing in terms of.
It is. But what I would suggest is no finger pointing. If I point one finger at anybody, 3 fingers are pointed at me. Sure, I can't point finger. Absolutely. The best thing we can each do is develop ourselves. Mm-hmm. So I don't know if Gandhiji said this or not. Be the change you want to see in the world. I mean, I think it's usually attributed to Gandhiji, but, but be the change in the world. That's the best thing we can do. And if you're not the change that we want to see in the world, we are hypocrites. We don't want to be hypocrites. That's it.
Absolutely. Any further questions, Dr. Bhoop? Anything you would like to discuss with Dr. Hussain for the benefit of the entire group here? Something that would be enlightening.
Yeah. So word of advice with respect to, because biologics, biotechnological products have a lot of future. And in your country, your erstwhile country, that's India, a lot of work on biotechnological products is being carried out. But as far as quality by intent or QBD, implementation of on biotechnological products is concerned, the scene is quite challenging still yet. So, what is your take? What is your advice? With respect to
Professor Boop, I was the president of biotechnology at a leading Indian company for less than a year. And after that, I dedicated my life to professional development. There's a reason for that.
Enough. 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 Thank you very much.
Thank you.
If there are no further questions.
Dr. Sharik,
Yes, sir, please.
I have one just observation.
Absolutely.
Dr. Azaz, he said, so before we point one finger to anybody, three fingers are pointing at us. And at the same time, that thumb, it is showing us that God is watching everything.
Yeah, exactly, right? Exactly.
So I appreciate, sir. You said it is all about higher level of consciousness. Now, unless the pharma industry and as a matter, anybody, unless we gain that higher level of consciousness, then we never think of the true human development. We always think of profit development. So that is really an eye-opener comment by you. Thank you very much.
One point of discussion, one more point of discussion. We hear a lot about ICHQ 14 for pharmaceutical analysis. So many meetings have been held. Earlier, it was solely dedicated to analytical quality by design. Now it is for changing the ICHQ to A as well. And since you are at the helm of the affairs in all these meetings, can we know about its recent updates with respect to implementation of QBD guidances for analytical development?
Right. So I think it's a very important and subtle thing. And I think what I would like you to bring to your awareness is FDA just signed an agreement with National Institute of Standards to progress further. Right. So what is the difference between National Institute of Standards and, say, Pharmacopoeia? I think that's the key. So see, our history has began with pharmacopoeias, where we needed to be able to test in the marketplace for quality. Right, that was the history. And by the way, in ICH, we have never been able to harmonize across the pharmacopoeias of the world. And so pharmacopoeias are market standards. When tested, you have to conform to that. But pharmacopoeias were never designed for quality control test, because if you ever read the USP correctly in the preface, you will see USP tests apply only to the units tested, not to the batch. That's the disclaimer. And the reason for that is those tests with pass-fail criteria were intended for a pharmacist or anybody else to be able to test the sample they have, not for the quality control. So, but it's a market standard. When tested, you have to conform to that. There's no, but you cannot aim in your quality target product profile and put in the USP monograph as your quality target product profile. You have to aim higher, and you have to phrase your specifications as not pass fail. No unit shall be outside 80 to 125. That's a zero tolerance criteria. That's not how you have a continuous variable. The 95% confidence interval of my blend uniformity or content uniformity is just not no unit shall be outside 80 to 125. Because by virtue of when you say that and you say 6.8% variability is acceptable, by definition, every batch is out of specification. It was by random sample that you found an out of specification or not. And we spin the wheels in, so you can never have continuous improvement when there's zero tolerance criteria. You can only have continuous improvement when you express that as a continuous variable and plus minus confidence interval. So you have to change the way you set specifications, right? So now for PAT and analytical validation, they brought in the same old concepts of HPLC or solution chemistry for when it comes to physical attributes, particle size, shape, and so forth, each is a unique. Dissolution, each is a, so dissolution calibrated tablet is one of the worst calibrated tablet on the market in terms of quality. And you calibrate your equipment based on one of the worst quality tablets. And how can you be better than that worst quality tablets? In fact, it was graduate students at University of Maryland who did that. I don't know whether Sharik would remember that. That was the whole story that I wrote for Mala Menon, is the editor of the Indian Dissolution. So the whole chapter I wrote for her. We have to get over that. So so ISTE Q14 is just a fix on all these unanswered questions. So it's not going to get to the solution. Sorry. I hope I answered that question. It's a big picture question, but that's how it's going to evolve.
Yeah. I got so many leads from here. Yeah. Right.
There's one question that coming out for Dr. Fayazuddin. He says, "So, what's your advice to Indian pharma fraternity post-COVID?" Because he can predict and compare India and USA context. Any thoughts on that?
Well, my heart is in India, so I think I've been engaged in a lot of training of students at the academia to industry with Mala Menon and my other colleagues from Bombay College of Pharmacy and other places, and especially with a young chap called Dharmesh Mehta in India. So what my learning there is, I think, onboarding of people, training and onboarding of people from academia or from other companies into your system is the door opener for building quality management system there. And so filling the gaps, acknowledging the gaps in our education and training and experience and professional development, and committing to professional development. So I am advising a company in India, I won't name it, and with a manufacturing plant in Goa. So I've been advising them almost pro bono since last 5 years, and they haven't received a single warning letter yet. And that was to corporate continuous improvement program and individual professional development program. You bring those hand in hand. So you essentially have a professional development program for each individual, and that goes hand in hand with corporate improvement programs, which will be required by, and in fact, FDA is running a pilot program on maturity of quality management system. So how can a quality management system, which is a socio-technical system, mature without maturity of the people who run that? So those pieces are coming together.
Right. So in terms of, we've even had a decent position in terms of leadership in generic providers to the globe, and moving into more of a biological era to continue to have that kind of leadership role, in more of we would say a biosimilar space, and related to that, what do you think are the critical limits? I don't think setting up an infrastructure would be a bottleneck. Do you think more of a technical expertise and bring that kind of core niche expertise would be a game changer?
So India perhaps has the most shiniest facilities, most up-to-date facilities in the world. Okay, so manufacturing facilities in India are second to none. Let me put it that way. Right. I think where we have to focus now is appreciation for system and process understanding and systems thinking. We cannot we cannot just focus on outcome and results, because we incentivize for results only. I don't care how you get that gas cylinder to me, get it at any cost. That's all I care about. That means we have to think about the process of getting to the outcome. Not just incentivize for results, incentivize for how you get to the results. I think that's the key, I would suggest.
Okay, I think that really makes sense, especially with biologics. There's so much of gritty nuances within that in terms of the whole process. I think so. To coming to that, I think that would really need, as you mentioned, a shift of mindset as such.
Yeah.
If we really want to continue our leadership into more into biologics areas. Professor Fayazuddin has a question interesting. He talks about how you relate vaccines potential and why Indian vaccines are not up to the mark. I don't know if you want to answer to that question. As such, he has concerns about Indian vaccine, Covaxin, and how you relate vaccine potentials.
Well, I think this 2-3 years from now, we will think about vaccines very differently. Let me put it that way.
Right.
I think I prefer a systems approach, not one size fits all approach. I want to make sure we focus on prevention. We focus on therapeutics. We have sacrificed therapeutics by design to promote vaccines, and that is something that will come back to haunt us. I guarantee you that. I think what I see of propaganda on vaccines is very troubling to me. And at the moment, with my grandchildren, I'm actually, this is going to cross a line. I'll be one opposing those damn things for my grandchildren. I think we have to make risk-based decisions. So I'm very emotional about that. So perhaps I don't have the right answer for that. But I would say within 2 or 3 years, we will have to learn from the mistakes we are making now.
Right. I think it's a very interesting discussion that goes on across. I mean, in country like US, which is so advanced, I mean, there are still debates about vaccine. I mean, people talk about Indian population. There, people are illiterate and they're scared about getting a vaccine. I think there is some concern.
Sorry. I will stop you there. People are wise to be afraid of vaccines.
Right. Okay. So I think that you've nailed it down essentially. So it is not just the ignorance as such. It is just maybe the distrust of the system somewhere around.
Absolutely. If you trust the system will kill you.
Right. So I see that you're fairly kind of.
No. Don't take me there. It's a very emotional topic.
That's fine. Okay, so I understand that. I think we are all going through that, and that debate comes on and off across. And when you answer people, there's a lot of skepticism around. And my relatives call me from my hometown.
It is, and I think having a scientific debate has been censored. We can't have a scientific debate on that. So that's the reason. The reason there's a good reason you don't trust the system when the system censors open scientific debate.
Absolutely. I think there is a lack of transparency. There's a lack of debate, and that kind of fuels the suspicion. And then people say, "Ki bhai, kya baat hai? Aap vaccine kyun lena cha rahe hain? Padhe likhe insaan."
No. So your words are saying as an excuse of fueling. Fueling suspicion is a good thing. We are supposed to be skeptic as scientists.
Right. I understand that. So if at all there are no further questions, I think it was a very nice and intense discussion. I think true to what the expectation was of a grand finale to our faculty development program. Thank you, sir. You kept to the mark, and I think we would like to continue to bother you and bring you more and often at these forums and share your passionate thoughts about things. I think that these are movers and drivers essentially, and it really provokes you. And I've been a avid fan of yours. I really go through your LinkedIn posts, which are sometimes I used to be cryptic, but I spend now a fair bit of time trying to understand them now. I'm getting a hang of it now.
You have to be cryptic to tell the truth, okay?
Absolutely.
You can't tell the truth anymore.
They're provocative, so I kind of spend our time now. I would encourage all of the participants out here. They should be following Dr. Ijaz for his fantastic LinkedIn. Sometimes can be a trash, but I follow his lot of posts, and they're fairly intriguing. They're provocative. They may force you to think about a lot of things. So I really thank you, sirs, for putting up those side. So if there are no further questions, I think I would like to invite Professor Abu Sufyan, a lead coordinator, for a closing vote of thanks. Over to you, Professor Abu Sufyan.
Thank you, sir. It's my honor and privilege to propose vote of thanks of this 6-day AICTE ISTE sponsor refresher program. I sincerely thanks to ISTE and AICTE for sponsoring this entire event. I thanks to Professor Vijay D. Vaidya, Executive Secretary of ISTE, for gracing this valedictory program in spite of his extremely busy schedule. Big thanks to the Dr. Bhupender Singh Bhup for his guidance and kind.
Professor Abu Sufyan, you're not audible.
Dr. Zeenat Iqbal from Hamdard University for their immense support and guidance. I would like to express my sincere thanks to my organizing committee members, Professor Maria Lal, Professor Madhuri Gaikwad, and Professor Salman Beg for being valued part of coordinating team and their enormous cooperation in organizing this event. As you know, event like this cannot happen overnight and it's required planning and a guidance. We have been fortunate enough to be backed by very motivated leader, our own president of Anjuman Islam, Dr. Zahirai Kazi, Honorable Executive Chairman BINM, Mr. Burhan Harris, Director of the campus, Dr. Abdul Razzaq Hanudgi, and Dean of School of Pharmacy, Dr. Sharik Sayyed. We thank you all for your valuable guidance and support. I also thanks to all the participants of Refresher program for your active participation. With this, we are done with our 1 week AICTE ISTE Refresher program. As per schedule and ISTE guideline, we have released 2 separate links, one for attendance, come at feedback, and second for quiz assessment link with MCQ based question for being eligible for the certificate. Have a nice day. Thanks for joining this program and being well-disciplined participant. Stay safe, stay healthy, and stay blessed. Thank you.
Thank you. Thank you, everybody. Thank you so much, Dr. Hussain. Thank you so much, Dr. Vaidya, Dr. Bhupinder Singh. It was an honor and pleasure to be hosting you. Thank you, Chairman sir. Thank you, Dr. Abdul Razak sir. Thank you. It was really a pleasure. Thank you, sir. Thank you all the participants. You've been a great participant for the entire week. Very diligent, very on the time. It was an honor and pleasure to be hosting all of you. We look forward for next exciting FDP from Kalseka Technical Campus. Thank you once again.
I'm keeping this meeting open for a few more minutes so that you can complete with your feedback and also you can give the quiz.